Good morning, everybody. For those that are physically present and those that are on a Zoom call, it's a pleasure welcoming you into the developing of the STEM workforce. And thank you for joining as we gather to look at the future and take stock of the past and make sure that we have all the ingredients to make this innovation conference and innovation research conference a springboard for what we want to look in the next, I wouldn't say 50 years, but definitely 20 years. When I hire people in my office, I always ask them, how do you want to look like 5 years from now? And 10 years from now? No one answers me 10 years. No one answers me 5 years, but I get answers for 3 years. So if we walk out of this conference and we have a path for 3 to 5 years, we're a winner over here. Last year, just as a recap, a historical recap, we celebrated here at Turo the 50 years anniversary, which is usually called by many people, and in the Bible, the Jubilee. And also, there was a time by which we wanted to take stock of the past 50 years, and we wanted to chart the future and see how this institution is going to look like 50 years from now, 25 years, 5 years from now. This year, we turn towards a topic that is very dear to me, the continuation of that great work with the continuation of research, and how do we nurture stronger, broader, and consistent presence of what the STEM career would look like. In essence, how are we going to nurture the next generation? The best answer to this question is in the spirit and the importance of the collaboration, which is going to remain multidisciplinary, interdisciplinary, and multifunctional. The disease that we're facing in healthcare are so complex that no one can pretend to come and say, I will attack this disease by myself and provide answer. That was when I started my career 30 years ago, and we could, because we were narrow-minded, we wanted to look at atherosclerosis as an example, and we look at lipid deposition in the artery. Little did we know that inflammation and the process of invasion of the intima by macrophage would be so critical into this aspect. Today, it is so complex that no one can come and pretend to address it. We come together, and the multidisciplinary aspect or interdisciplinary aspect is crucial if we want to be successful for the determination and making sure that we have a program that is going to address this tailored medicine that we all look for, this pharmacogenomic that we all look for, those concepts that are so specific and appropriate for each and every patient that we have. We will introduce, and I would be participating to this program today, but I'd like to acknowledge our collaborators, Jain here, the co-founder or the director of Westchester Biotech Project. Thank you for all this hard work, and I appreciate it very much. My office has been also, I haven't done anything. Paola has done everything for this, and at time I did ask her, what was she doing really? And I see that what she was doing made sense. The WBP is an organization that creates opportunities for the collaboration across borders, silos, and scientific culture. Definitely the mode of operation of this institution and the place where you are, the incubator that we have. Together, we will be introducing and moderating a group of speakers of the STEM force. These professionals include Supriya Jain, which I have had the pleasure of interacting with her in the past from the Maria Ferry Children's Hospital, the dean of our school, the Mary Laurie, of the Touro University School of Business, called also the Win Thurlow Executive Director of the Life Science New York, and some returning familiar faces that we will introduce as we move on. René Pereira, our dean of the school that we just created recently, Touro, has opened a new school of medicine and osteopathic school. She heads the Office of Research. I am very pleased to see you this morning and for you to participate here. She heads also a major operation of research, McLaughlin Research Institute over there, and we are in discussion to make this institution a stronger research environment. Without further ado, I think I can say that I'm extremely positive that we will have and we will all gain a tremendous amount of information by the panel. Thanks to the Westchester Biotech Project for collaborating with BioInc. And with that, I will turn it to Joanne so that we can get the program moving. Thank you.
Thank you for your kind words. Really appreciate it. This has been very fun for us. You're going to hear from Paola in a little while. We have a couple of game day changes, like always. One of our speakers, Win Thurlow from Life Sciences New York, got sent home because they've closed down Route 17 from Binghamton down because of the snow. So I assured him there's a lot of big talkers in this room and we'll have no trouble filling his time. So I'm sure we'll do great. Here we go. Glasses or no glasses. I'm just going to share a little bit about us. So Westchester Biotech Project, my co-founder, Michael Willing, and I started talking in 2015. And the women in the room will appreciate this. He called me on the phone. He said, "Do you want to do," oh, is there, not this? Okay, thank you. He got in touch with me. He said, I was working for another organization. He said, "I see what you're doing. You want to try it in Westchester." And then he said, "I'm going to call you back in 2 weeks." And he called me back in 2 weeks. And so I'm looking for nods, right? It's a good person. Anyway, Michael's son, who you're going to hear more about later today, is just about ready to go to college. And this young man had to have 2 stem cell transplants as a baby. So when the family got through that nightmare, he wanted to figure out a way to give back. He started talking a lot more with scientists and doctors, and is a native of Westchester, grew up in the area, and lives in White Plains right now. And my experience, we all have family members. My mother had Wegener's syndrome, which had a huge effect on her. She lived into her 90s, which was amazing, but it certainly had a big effect on her. And so, and I so appreciate your comments, Dr. Amar, because it is all about finding unlikely connections across all the different silos. We found out that if we started to open the door and gather people together, people would come together. And we've had over 100, almost 125 registered sessions since then. And through all that, we've done a lot of listening. We've focused in different areas, some of them more broad business type topics, education topics, scientific topics. We have a whole consortium around the microbiome. We have one around clinical research. So there's sort of, as people found each other and liked the environment that we have helped to support, they started to say, well, let's do this, let's do that. And I would say, great, you organize it, and off we go. We started off with a concept to think about the area in terms of opportunity density. And if you go to Boston, I know a lot of you have had a Boston experience. If you go to Boston to work for a startup and your company blows up, you can cross the street and find a job, give or take. And there's, what do we do to enhance the environment here so there's more comfort for companies to come here, for companies to grow here, and for people to come and work here. Here, I've just pulled out 4 things that we learned through this whole process. Kids talk themselves out of science and math in middle school. Okay, I'm one of those. Young investigators. which can be the whole pipeline. I think about it as a sort of PhD postdoc era through around age 40. You're finally out of school, you're working, you may well be trying to start a family, and you're creating influence in your career that's going to make you more and more powerful later on. And so to me, this is a really important phase of a scientist's life to give support to. This is what we're going to be talking about a lot today. We're here in a lovely incubator, and at lunchtime, please plan to take a tour. Paola's going to walk us around. What happens when it's time to leave the incubator, and where do you have to go? You're going to hear a real true life story about a graduate of this incubator and the process they went through to grow and find another place to be. My next step is that, so I'm working on retiring. And my next step is going to be to work with Life Sciences New York on a statewide initiative to build more expansion stage labs around the state. That includes Westchester, but it's Buffalo too. And finally, everything exists. The knowledge is here, the money is here, all of the know-how and resources are here. And a lot of what we're going to be talking about today is how do you actually bring the right people together to make things happen. We have some of the people who have made huge projects happen in different ways. We have the story from a company that's a real company right here in Westchester. That's a perfect example. And as we get through the day, at the end of the day, we're going to have a round table and really invite everybody to speak. But we'll want to think about, okay, now that we know each other better, how can we work more closely together to make things happen? And yes, the question is, would you come together? Would you come and talk with us about your science? And the answer has been yes. Our first panel is going to talk all about this particular phase. And would the panelists come up, please? And Paola, would you come up? So Wynn Thurlow is the person we won't be seeing, but we have Ofer and Nancy and Leela.
All right, hello. So first of all, I would like to apologize once again. As we mentioned earlier, there have been some traffic delays with some of the commuting trails over here. So we have much fewer accompaniments than we would have liked. And a couple of our speakers are coming in a bit late as well. But we do still have a very good lineup for you. And that begins with a small introduction. Some of you know me, some of you do not just yet. My name is Paola. I am the coordinator for BioInc at New York Medical College, which, as Joanne here mentioned, is a biotech incubator that focuses on helping startup companies at early and mid-stage levels to really get what they need to succeed in the environment that they are in, whether they are trying to pursue therapeutic tracks or some new technology that they could provide to hospitals, sometimes to dental schools, or even our own academia. From there, what we do is we provide the space, we provide the resources.
You while we're fussing with slides. They don't want to watch us.
So what we do here is we provide the resources, we provide the networking and the space really that they require. And it can range from making sure that they have the exact equipment that they need for their research to be conducted to connections, right? To investors, to potential liaisons that they could be collaborating with. If they're stuck on an idea in their research, someone that they can talk to get out of that rut. And really it ranges from anything beyond that and above. What we have been trying to do since then is that whenever I go to these similar conferences to what we have today, one of the topics that has come up predominantly is that once companies have reached a certain level, their next goal is they want to give back. They want to get involved with generating that next generation of the STEM workforce. And how do they do that? Some of them end up volunteering, and then you've got Regeneron, which produces these competitions for students at high school levels, at college levels, and it ranges, right? But the question is always, how do we get people invested in STEM careers and to continue in them? So as someone who started in STEM as well, I personally am very passionate about it on a personal level, on a cultural level, and of course on a business level. This, for me, is personal. And so when I saw that this is something that our entrepreneurs are wanting to reach back into, I was more than happy to help tap into it. So again, we are based on a medical campus, which means we have the means and the resources, once more, to connect students to that next level of passion for a career in. And it could be on the business side of it, as we'll see from some of our speakers here today, and it can be on the direct research side of things. There are my slides. So some of this I've already said. But so again, a brief introduction to BioInc. What we do, again, is we focus on helping startups to really make their grounding through different levels, whether it is at a local sphere or abroad once they have um, the means, the patentings, everything that they are required to succeed. The brief introduction, which again I already gave, so I won't repeat myself for you. The overall goal that we have in the Mid-Hudson region and really for New York State as a whole is that we wanted to answer the question, why are we not also an innovation hub, right? Normally, when you think of innovation hubs, you're hearing about Silicon Valley, you're hearing about MIT and all these other universities in Boston, but this is New York. We also have a lot to offer here. We have a huge hub of researchers, of pharmaceutical companies, science centers, but the real issue is that we are not connected. So our main goal from a lot of the organizations that you'll hear from today is to bridge those connections and to really leverage what we have available in New York so that we can garner more researchers into the area and keep them here. So with BioInc directly, we have helped 13 companies grow over the last 10 years almost that we have been in standing, which has led to over 90 jobs being created and over 130 million in fundraising that each of these companies has been able to produce altogether. The impact beyond that, though, is that there, again, it's a building of collaboration efforts. There are some of our companies end up still working with the college or with the hospitals that we are affiliated with and continue to use the resources that we have provided here, whether it's our research cores that have a strong backing in genomic research or in our animal facilities, or even just our patenting office. So we do provide a lot for our companies even after they have graduated from within our walls. And again, this reaches back into the overarching goal that we have in the New York State, which is to further develop the life sciences community that we have here. Now, at the college and within the organizations that you'll be hearing from today, that goal is reached in different ways. So some of our deans here, for example, focus on getting women into STEM careers. They'll focus on developing the business school in a way that, can connect to research. Because a lot of times, one thing that I've learned with our students here is that people can be a bit focused, right? You come here for a degree in research or you come here for a degree in business or some level within that, and you don't quite look beyond that other than what is directly in front of you. So my job is to help broaden that perspective, right? So we have the business school coming through, we have the students at some of our other affiliated colleges learning how they can bridge what they are researching or studying for, whether it's in marketing or in business management, and how can they apply it to a healthcare level, to an entrepreneurial level. And that is, at the end of the day, our goal, to help develop that at a nascent level, as well as in the companies that we are working with directly. The way that we have started doing that ourselves is to get tours through some of the high schools and colleges that we are working with so that they learn about different careers that they can pursue in STEM through tours that we provide to them here at New York Medical College. And our ultimate goal later on is that we want to broaden this. We want it to be inclusive to really any of the innovation centers that we have in the Westchester region. The second way that we do this is, again, we start working with like-minded organizations such as Sister to Sister, Builds Bio, and many others that, again, you will hear from today. The final way that we do this is, at the end of the day, we are still an incubator, and our focus is still to try and help these nascent companies strive in a very competitive arena. So we've done that by continuously graduating our clients, and when we have the capacity for it, we also like to expand the number of labs that we have available to them. So in total, BioInc right now has about 1,700 square feet of office space, lab space, and collaborative spaces that can be used by these companies. And that's only within BioInc's walls. Beyond that, again, we do still have the companies collaborating with our different research cores, the animal facilities, and affiliated institutions. So, sorry? Yes. Did I say so? Ooh, yeah, that's a bit different. Yeah, 17,000, sorry. In conclusion, I would like to leave us with a quote that is my personal favorite since I was in high school, and I think it's very apt for what we are discussing today, which is, in times of change, learners will inherit the earth, while the learned find themselves beautifully equipped for a world that no longer exists. And I think in science, in general, we know this to be a fact, right? What you knew 10, 15 years ago is not the same to what we know today, not the same to what is available to us today, whether it is in the companies or in what we know for our health, for the world around us. And with that, I would now like to introduce our panel. So for our panel in the first team that we have today, this is for what we call Blueprint 2030. And it is focused on the companies or organizations that are either directly benefiting from organizations that focus on building out for entrepreneurs, meaning it could be in real estate, it could be companies that, I'm sorry, organizations that aid in connecting these entrepreneurs to each other, to investors, to others that would be beneficial to their growth, or the companies themselves. So I will give a brief introduction and then let them talk about what they do. So the first up, we have Dr. Laila Gamsari, who is the Senior Director of Discovery Sciences at Sapiens Therapeutics, which is one of our recently graduated clients. They focus on developing peptide therapeutics for cancer patients, and they are a clinical stage biotech company. Dr. Gamsari is the person who is pioneering the discovery of these therapeutics in vitro pharmacology and in difficult to drug targets in various cancer types. Then we have the lovely Nancy Kelly, who is the founder and CEO of the Nancy J. Kelly and Associates organization, which then leads into Builds Bio. Now, formerly, Builds Bio had a sort of three-part name to it because they work with 3 different states, but now they've kind of merged all together because, again, the goal is to create a collaborative nature and a stronghold in this region. So Nancy's organization focuses on really building and nurturing these key strategic initiatives in the New York region, well, now beyond, that focus on developing our life sciences community. And to name a few of the organizations that she has helped to build up, we have the New York Genomics Center and the East River Science Park, as well as some other projects that I'm sure she'll talk about today. So I will leave the rest of that to her. And of course, Mr. Thurlow, who is not with us today. And then we have Ofer Ohad. I hope I'm pronouncing that correctly. Okay, great. He is the managing partner at DPI Projects, which is a company that brings more than 2 decades of real estate expertise to capital projects, including, of course, tech startups, locations that are similar to BioInc but on a bigger scale, that provide space for over 250 companies and over 1,000 entrepreneurs. So with that said, I will let them now take over and give their introductions.
Good morning, everybody. My name is Laila Kamsari. I'm a senior director at Sapiens Therapeutics. And I essentially lead the discovery efforts at Sapiens to discover peptide therapeutics.
My name is Ofer Hall, as you heard. I'm with DBI Projects. We'll show you a little bit about that later on. But we're essentially assisting companies in developing facilities and combined hubs for both technology, life science, and others.
Hi, I'm Nancy Kelly, and I'm the CEO of Nancy J. Kelly and Associates, and a founding member of Builds Bio, as Paola said. We build things that matter for science and medicine, and I'll talk a little bit more about that in a few minutes.
Good morning, everyone. It's a true privilege to be here today. I'm honored to represent Sapiens and tell you our story of innovation and service to cancer patients.
You have to scroll.
Okay, I figured it out. We are a clinical stage oncology company focused on difficult to drug targets, as we heard from Paola. We develop spares. Those are stabilized peptides engineered against regulation to target difficult to drug cancer targets. Those are generally protein complexes, such as transcription factors that reside within cells. And for any drug company, one of the biggest milestones is to transition from preclinical stage to a clinical stage company. For us, this happened in 2020 with the development of ST-101, which is a peptide spare that targets one of the most difficult and aggressive type of tumors, glioblastoma. It's a type of very aggressive brain tumor type. Again, this year, we entered into the clinic with our second peptide, ST-316, which targets one of the most elusive cancer targets, beta-catenin Wnt pathway. We have 8 discovery stage spares in our pipeline, and we have a platform that we developed this year, and it enables us to discover more spares. When we speak about difficult to drug cancer targets, it's not because of the nature of these proteins. It's really because of the limitations of the tools that we have in our hands. Currently, biopharma companies develop 2 classes of molecules. Small molecules might be more familiar to you. Those are chemical structures that can easily travel across your body and can penetrate easily into cells. And they bind to small molecule pockets on the surface of the proteins and target them. However, not every protein in the cell has those small molecule pockets. And therefore, biotech companies have developed larger molecules, such as antibodies, that can bind to the flat surfaces on these proteins. However, they are too large to penetrate into the cells. Mostly, they target proteins on the surface of the cells. We develop spares. Those are miniaturized proteins that can bind to these flat surfaces on the proteins. They are small enough to get into the cells easily, and they are at the same time large enough to bind to these flat surfaces. They're generally not immunogenic, one of the major issues that might exist for drug development efforts. Let me now share with you a little bit more about ST-101, which is our lead peptide candidate in the clinical trials. This peptide targets a previously undruggable cancer target, CBP-beta protein. CBP-beta is a master regulator in glioblastoma, and its high expression level is generally correlated with lower survival in these cancer patients. The first question for us was if ST-101 as a peptide can cross the blood-brain barrier and reach to the tumor sites in the brain? And if it does, can it bind to its target, and what it happens to its target? Since we have access to biopsies from the patients in the clinic, we took the biopsy from the brain tumor. And using a method called immunohistochemistry, we stain for the presence of ST101 as well as its target, CBP beta. If you look at the top two panels, top two pictures, those are showing the sections of the tumor stained for the presence of ST101 post-treatment. Each blue dot represents a cell. And ST101 is shown with the brown color. That's an antibody that we can use to stain this peptide. And you can see that not only ST101 has crossed the blood-brain barrier, but also it has penetrated into the tumor tissue. At the same time, we asked what happens to its target, CBP beta. If you look at the two bottom pictures, it's staining with immunohistochemistry for the presence of CBP beta pre-treatment and post-treatment with ST101. And as you can see, CBP beta level has significantly reduced post-treatment. This is quite consistent with the mechanism of action of the peptide that we published last year. ST101, after binding to its target, that directs it to degradation. Now I would like to share with you one of our cases in the clinical trials. This is a 34-year-old male patient from New York. The patient was diagnosed with glioblastoma in June of 2021. His tumor was removed, and he received radiotherapy as well as chemotherapy. His disease recurred in November of 2021, and the patient underwent a second resection. We enrolled this patient in January of 2022 in our ST101 clinical trial. In the images, you're looking at the MRI images from the brain of this patient. The green arrows represent the dimensions of the tumor. And as you can see, only 4 months after receiving ST101, the size of his tumor was reduced to 50%. And in the following 7 months, the tumor completely disappeared from these images. What we hear from the doctor that treated this patient is that the patient was free from his clinical symptoms, and the patient went back to normal life. Unfortunately, this patient developed a new lesion in February of this year and was taken off the study. Nevertheless, ST101 continues to benefit cancer patients, GBM patients in the clinic. We have a few patients with positive response. I'm really excited to share this news with you. And there are multiple patients that their disease have been stabilized. We are still studying ST101 in clinical trial with recurrent glioblastoma as well as newly diagnosed glioblastoma. Our second peptide, ST316, as I mentioned at the beginning of my talk, is now in the clinic. And we hope that by the middle of the next year, we start phase 2 clinical trial to treat patients with colorectal cancer. Our third peptide, June AP, is in line to get into the clinic. And we have 7 more targets, previously undruggable cancer targets in our pipeline. And we continue to innovate and bring options for cancer patients in the clinic. I hope next year I get a chance to update you on the progress. But with that, I would like to thank Joanne, Paola, Dr. Amar, and the organizers of this meeting. And I'm happy to take any questions you might have. That's a great question. We actually have an active domain for our peptide that can disrupt certain protein-protein interaction. Those are transcriptional complex protein interactions. We attach to that a penetration domain that direct the peptide to the cellular compartments where they are supposed to be active. For example, if it is within the nucleus, we have a penetration domain that enables the cargo, which is the active domain, to get into the nucleus. At the same time, we have assays and methodologies that enable us to look for the exact protein-protein interaction that is supposed to be disrupted, right? We have multiple cell-free and in-cell methods that enable us to see that disruption. And as a result, you expect to see a reduction in the level of target genes. And we have qPCR methodologies, and we have reporter assays that enable us to answer those questions. Sure. So in the preclinical studies, we know that most of these transcription factor levels are higher in the tumor compared to normal cells. And we know that because they are at high level in the tumor, we are affecting them more. But you're right, there are normal cells that need these transcription factors for their normal function. We do toxicology studies preclinically, and within the clinic, we look for adverse events, and we record them. So far, we haven't seen adverse events for ST101. There was an adverse event that we control with antihistamine in the clinic. But generally, yes, you're right. We at every stage, in the discovery, in the preclinical stage, in animal studies, and in the clinic, we look for those adverse events and we record them. But so far for ST101, the histamine production has been the only one that we have seen. Sure.
Yeah, thank you very much. And thank you, Joanne and Paola and Dr. Amar, for having me here this morning. I'm really excited about what's happening in the greater New York metro area right now, which includes the whole state of New York, Connecticut, and New Jersey. We are literally the largest aggregation of life science activity in the world. It's the largest life science economy in the world, and it's a very exciting place to live and work if you are in science right now. Dr. Amir was talking about 3 years, 5 years, 10 years. I want to tell you what two of the top innovators in the last century say about the next 100 years, which is the century that we're living in right now. Greg Venter, who was part of the Human Genome Project, said, if the 20th century was the century of physics, the 21st century will be the century of biology. And Steve Jobs followed, the biggest innovations of the 21st century will be at the intersection of biology and technology. A new era is beginning. I basically presented these slides last year at Toro's 50th anniversary, but they're no less true this year. And as science and technological change accelerates, we are really seeing the beginning of a fourth industrial revolution in which the lines between biology, physics, and the digital realm is blurring. And as Dr. Amir said, the multidisciplinary nature of science is allowing us to make discoveries that were not possible before when scientists were just working singly or individually. And what this means is that science in healthcare and the life science market in general is changing and evolving faster than ever. And what's driving it? Basically, technological change. So you have the Human Genome Project, which, sequenced the first human genome. It cost $3 billion, and it took 15 years to do it. Today, we can sequence a single genome in under $1,000 in less than 24 hours. That's what's driving the pace of progression and advancement in science and medicine today. So it's about sequencing. Now that we have discovered the basic protein building blocks of DNA, which basically is 4 building blocks, A, C, G, T. If you think of zeros and ones, which are the binary bits by which we write software code, we're now writing the code of life, the biological code of life, where scientists can basically design new biological products in silico on their computer and then boot them up in their labs to see if they're going to behave the way that they expect them to be. All of this activity is creating a huge amount of computational data, big data sources, bases that need to be mined for patterns, and algorithms are doing that today. Artificial intelligence is finding those patterns and allowing them to use the patterns faster and faster. And then, of course, new tools and software that are also being developed, like CRISPR, for example, the first therapeutic to treat sickle cell disease with a CRISPR therapeutic was just approved in the United Kingdom for the first time. So that was last week, I think, which is very, very exciting. And this is just a graph which shows you the reduction in costs in sequencing the human genome from the end of the Human Genome Project until 2021, which was just a couple of years ago. And so what we've got today is we really have a bioeconomy which has developed, which is being fueled by biology and technology, just as Steve Jobs said. And we're using that to create new food, new materials, new fuels, all kinds of new drugs. And it's changing the way we understand our bodies and our nutrition and our health, as Paola just said. So what does that mean for New York? Well, New York, as I just said, is the center of the universe for life sciences in the globe today. And really, all of this started with the creation of the East River Science Park, which is now the Alexander Life Sciences Center in Manhattan, in the early 2000s. And when I first walked the site where the Alexander Life Sciences Center, which is 2 very large, beautiful buildings today, now stand, there was an abandoned laundry building filled with asbestos on the site. And you walked beyond that over a crumbling street that was on the other side was the Bellevue Psychiatric Hospital. That was a homeless shelter. And beyond that was a parking lot and a tent which housed the unidentified remains of the 9/11 victims. That's what the site looked like when I first walked it. And today, what you see is a 3.5-acre park and 2 towers, life science towers, which were built and housed the first speculative life science companies, pharma and biotech companies, to come to New York City and locate there. That was in 2006 and 7 when we were building and developing that project. 3 years later, I founded the New York Genome Center, which is a high throughput sequencing facility in New York City. That's at 101 Avenue of the Americas. And in order to create that, it had a physical requirement of 173,000 square feet. We converted the first office building in Manhattan into a lab building. And it was the first time that conversion had been done in Manhattan. And so those 2 developments really fueled the creation of what is now happening in New York. This really got accelerated in 2018 when Mayor de Blasio, mayor of New York City at that time, announced $500 million in investment in the life science industry, followed closely by Governor Cuomo's announcement of about $600 million investment at the same time. And what this did This was the same year that Builds Bio was actually founded, which is an organization that brings the life science community scientists together with real estate and design professionals to create new space. Because in 2018, there was literally no private space available except Alexandria and the New York Genome Center. Those were the only two things in New York. And this is the way New York looks today. It's a very, very different place. In just under 10 years' time, over 3 million square feet in new lab space has been built in a variety of clusters around the city. So each one of these large yellow circles that you see is a life science subcluster that has developed either around a major academic center or around a major life science development like the Alexandria Center. So you see the arrow in the upper right-hand corner, that's pointing towards the Bronx, Tarrytown, and Westchester County, which is where we are today. You come a little south on the west side, that's Columbia Presbyterian, where Columbia's medical school is and where the Alexandria Launch Lab is. And then Manhattanville, where the Manhattanville Factory District and Columbia's Morningside campus has been located. And then you come down a little bit and there's Mount Sinai and all of the work that's going on around that in artificial intelligence. And then you come down that to where the academic alley is, which includes Rockefeller and New York Presbyterian and Cornell and Sloan Kettering, and all of the large academic institutions. There's a bridge on Roosevelt Island, Cornell Technion, which brings you to Long Island City, where there are new developments developing there as well. Inno Labs and Alexandria have some developments there, as well as Longfellow. And then you come back to Manhattan a little bit further south, that's Midtown East, where Alexandria is located, the Cure is located, the city and state have just announced the Spark Kips Bay project there, which kicked off yesterday in the EDC's offices, and I was there. Very exciting, $3 billion project that's going to be built over the next 10 years. And then a little bit south, and that's where you see Hudson Square, where BioLabs and the New York Genome Center is. And then you have a split south to Brooklyn and also to Jersey City on the southwest there. So all of these subclusters are basically growing now. They're growing in number of square footage, they're growing in number of companies, they're growing in scientific activity that's taking place there, and they're starting to be connected. So what you see in that big yellow arrow is what we call the BioArc, which connects all of these life science clusters. So just to show you visually what some of this looks like, walking down the East Side corridor, starting with the area around Mount Sinai, Elevate Research Properties in Taconic has just announced Iron Horse Labs, which is named after Lou Gehrig, who we all know was a baseball player that developed ALS and dedicated to his memory. And then Academic Alley, where all the large academic institutions are. This is Rockefeller's brand new campus that has been built over FDR Drive, and they have just established the Ford Center for Life Science Innovation, which will be graduating about 25 new companies every year. Over in Long Island City, we had the opening of Inno Labs, which has the pandemic response lab for New York City, as well as several of NYU's labs there. And that's King Street Properties. Here's one of the Alexandria towers that I was just talking about in Midtown East. This is the New York Genome Center at 101 Avenue of the Americas in Hudson Square. And in Brooklyn, this I think is indicative of what's happening. So each of those clusters that I just talked to you about has either one or multiple incubators that are now graduating between 20 and 25 companies a year. Now, if you add up all of those clusters and you add up all of these accelerators, incubators, just in the Brooklyn area, you've got a couple hundred companies a year which are graduating and growing and building this ecosystem. And even though those companies right now, when they get out of the incubators, they need 7 to 10,000 square feet of space. When they grow, they need 15 to 25,000 and then 40 to 50,000. And I think this is indicative of this slide because you can see the way it's happening in Brooklyn. So if you look at BioBat, which is on the far right here in Sunset Park at the Brooklyn Army Terminal, there's an incubator there. They are just a short way from Industry City, where there's a new incubator opening called Colony Labs. Their launch is on December 7th next week for biomaterials companies. SUNY Downstate is connected to both of these places. You can see that. They have 2 incubators at the apex of the subway lines there. And then NYU Tandon School of Engineering and their Urban Future Lab connected to the Brooklyn Navy Yard where New Labs is and Yard Labs and Dumbo and the new climate change accelerator that's just been established on Governor's Island. And then across the harbor and the river, you see SciTech City and EdgeWorks in Jersey City. And the Cove, which will be a 3 million square foot mixed use development there. But on the left-hand side, you can see all of those different accelerators, 1, 2, 3, 10 of them, which will be graduating these 20 to 25 companies. So that's 250 companies a year coming out of Brooklyn alone right now. So you can see the accelerating pace of this. And it took a long time to grow and develop. And so when people in Westchester say to me, the pace is slow, I say, it's coming. It's coming. And Sapiens is a perfect example of that. So on the west side, this is one of the most exciting new developments in New York City today, the expansion of the Hutchinson Metro Center, done by Simone Development Company. The Bronx has the most interesting combination of scientific institutions in the city. They have the New York Botanical Garden, where many of us forget that many of the prior pharmaceutical products came from botanical compounds in the first place. Any of you remember Medicine Man, that crazy researcher from the University of Pennsylvania running around the jungle trying to find an antidote for cancer? That's where many of our therapeutics came from. And the New York Botanical Garden has just established its first research arm and hired a chief scientific officer. The Wildlife Conservation Society, which has research collaborations all over the world on climate change and in a number of other areas around animal health, works with Cornell. The Bronx Zoo is the second largest attraction for international visitors in New York every year. We have Montefiore, which is the second largest health system in the state, and Montefiore Albert Einstein Medical School, which is number 40 in the country in terms of medical schools and is rising in the ranks. So lots of really interesting science going on there. A little bit further south is the Morningside, the Columbia Presbyterian around the medical school, which just created launch labs last year. And then in Manhattanville, you have the Tasty Building, which is a ground up development by Janus Properties, but there's also the Mink Building there. There's a Sweets Building. This was a candy manufacturing area in Harlem. And it is now a prime site for 21st century research and development. In Midtown West, we've got the Hudson Research Center, which is full at the moment. Again, Elevate Research Properties and Taconic developed this, but they have companies like C16 and Hybercell and Rensselaer Polytechnic Institute and the New York Stem Cell Foundation in there. And there's a real hub developing. And then in Jersey City, this is the 100,000 square foot new incubator being built by the Liberty Science Center called SciTech City. And this stands adjacent to a K-12 STEM school that is being established by Hudson County, Jersey City, and Liberty Science Center to train the scientists of the future. So this is really all so exciting. And I hope it gives you some idea of the robustness and vitality of the growth in this market. Thank you.
On? All right. Thank you all. Thank you for having us. Today, I'll tell you as a perfect continuation to Nancy's presentation about our company and a case study that appeared on your map and on your survey. DBI Projects, we are a development management firm. We're based here in New York City. We have also offices in Boston and Philadelphia and Austin, Texas. We're about a team of 65 people, and we've been around for just over 20 years, building close to 1,000 projects over different disciplines and practices. In essence, we both provide what we call planning and advisory services. We work with organizations to develop their programs, their strategic planning, building their budgets and schedules and everything that has to do in preparation for execution of a project. And then we obviously work with them in executing a project, overseeing it and making sure it's being delivered the right way. The case study I wanted to share with you is a new lab in the Brooklyn Navy Yard. This is our project back from 2016, and there's a chapter, the second chapter for it. Essentially, New Lab is a technology hub. It was established about 8 years ago, and it is a home for more than 250 startups that are in later stages of their growth. Some of the firms already graduated from New Lab and moved on, but New Lab has few kind of main themes of focus, including energy, mobility, urban tech, and the kind of the most interesting aspect of it from our end was that the New Lab started back when a facility was identified in the Navy Yard. It was a deserted ship manufacturing building that was used back in the 30s and 40s, all the way through the 50s, to build a big Navy vessels, which you can see on the side there. The facility was deserted then and stood about to fall apart for close to 30 years when it was discovered. And the idea was to develop this hub for different companies and facilitate them, allow them to use joint laboratories, joint shops, where perhaps an individual firm would not be able to support, but together they'll be able to utilize those facilities. Along the advantage of being able to collaborate and work together and share information and be able to feed each other with ideas. The facility was built and opened back in 2016. In early stages, there was a long list of firms that tried to get in, and there's still a long list of firms. And as a result of that, there was an extension that was built later on. The next phase of New Lab just opened in Detroit, and I'll show you in a second. But the lesson from New Lab is it takes a lot of effort and multiple layers of participation of different groups to make it happen. In the case of the Navy Yard, this was a joint venture between the private sector that invested equity in the venture, raised debt from conventional debt and more kind of creative debt through the EB5 program, as well as quite a few grants from the state level and the city level that allowed this whole venture to come to life. Obviously, a pretty robust legal organization had to be created around that. The next chapter I was describing was the new lab in Michigan in sponsorship with the Ford company. The Detroit facility is much larger. Brooklyn one is about 80,000 square feet. In Detroit is almost triple the size. It has multiple firms focusing again on R&D with an emphasis of firms that specialize in the car industry that are relevant for the car industry. Right now I can share that there are quite a few initiatives surrounding the next new labs. There are at least 3 of them that are being contemplated in the US and around the world. So that's my presentation. Thank you.
Bless Nancy. The last time she spoke for us was February 27th, 2020. There was a fire drill in the middle of her talk, and she came back for more. So thank you for that. And she's the Mac Daddy of getting stuff done in Manhattan. She's been involved with everyone. Yes, yes. And so she's clearly identified the need. More than 200 companies a year coming out of incubators in the New York City schmear, okay? How
That's just
do we?
Brooklyn.
You like that? I learned that a while ago. You got to do this too. And so what do we do to stimulate the next stage? DBI is the company that knows how to do it, which is great. So I love you being here and telling that story because all those partners have to come together to make that happen. So I'm going to ask you two to think about something while I put Leela on the spot. Is that from what you know today, what would you identify as kind of the opportunity? X number of square feet are going to be needed. If we today walk out and say, okay, we're going to build something, do we need $100 million? Can we start with $20 million? Where's a place that, and it keeps changing, but if you were to start today from what we have today, what would you see to make something happen in 5 years? Okay, so I'll let you play around with that. So Leela, I would really like you to talk about the process that Sapiens went through to find your place, get it approved, fit it out, and everything.
Thank you. Yeah, I was just sharing with Joanne before starting this panel that I walked into an empty lab in 2018, searching for a job, doing interview with Sapiens. And when I was told that you're going to work here, I was really surprised because there was essentially no lab. There were 2 empty benches, although the company was running animal studies behind us at BioInc Biovarium still. And well, I walked into that lab and started with the senior leadership, gradually building the lab. And at the time, we had ST101 developed already as a molecule. And we started testing that and understanding its mode of action and continue at the same time on testing in animals and proving its safety in animals, preclinical studies. And over the course of 2 years, we discovered the mode of action of the peptide that was eye-opening to understand our molecules better and to understand what it does when it gets into the body. And at the same time, we had other previously undruggable cancer targets in our pipeline that we were working on the discovery aspects of those programs, for example, ST316. At the same time, we had a parent molecule to target Wnt beta-catenin pathway. Wnt pathway is generally a pathway that has been the subject of decades of research in both academia and industry. And sure. Of course. Right. So I think at the time, the senior leadership team were looking for a space, and this was, I believe, at the middle of the pandemic, and there were not that many spaces available in Westchester to move to. I mean, there were plenty of office spaces, obviously, but lab spaces were limited. And at the time, I think that there were two spaces that were offered to us, and one of them was, I think, was a little bit larger, and at the same time, it didn't work for us. So we were first debating between that space and the current space. We are in Tarrytown. We are currently located in the BASF building, which is one of the major hubs of the BASF company, the chemical company. And there was a lab space empty. It's, I believe, is 12,000 square feet lab space that we currently have contracted to move to. And that's how we found the space. And we moved from BioInc. We graduated at the beginning of actually this year, end of 2022. We were just ready to move that to that new lab. And January of 2023, this year, we started working and building that lab. And since our space was much larger, we started to get new instruments. And then the office space, because our office spaces were located in Mamaroneck, we wanted all of us to get together from the office, people that were working in the office and people that were working in the lab. So it worked out for us that there was another building within the same BASF campus across the parking lot that is now our offices. Yeah, that's how.
Thank you. And that was lucky you found an existing lab so you didn't have to qualify a brand new lab.
Sure. Yeah.
So, I'm going to ask you, so from today forward, we want to see something in 5 years. How much money do we need and how big should it be? And I know everything will change, but just to give us a sense of the scale.
Yeah, I would say even before throwing numbers at you is that the world of facilities and real estate and spaces is going through a huge transformation, right? The more conventional real estate, the office and retail is transitioning into a new era. And while the space-making business is becoming more and more expensive, as construction is becoming more expensive, interest rates are high, the availability of labor and material is constrained. All of those are pressure points against the ability to create new space. The fact that the conventional real estate, offices, and retail are transitioning and in a crisis mode creates a huge opportunity. And while lab space is not available out there, ready to be picked, it can be created in environments that only 5 years ago were unthinkable. To think about beautiful life science facilities in Midtown Manhattan was unthinkable. To think that you can create R&D facilities were just silly in the environment of 5-10 years ago. Today, there's a lot of opportunities. There are landlords and there are developers just looking for academic uses, for R&D uses, for life science. And while cities like Boston and the entire Boston metro area starts to sense some saturation, New York is starving for that side.
So you think the opportunity is in converting office space?
This is a great opportunity, especially in the older older facilities, older buildings that are no longer conducive to modern office use. They're no longer conducive to conversion to residential uses. These are where great things can happen in a
So
very
let's say we
affordable
found a
basis.
if we found a 50,000 square foot office building to start with, right? Somebody gave us a building. Now what do we have to do? How much do we have to raise to turn
Now
that
you
into
have to
something?
find $500 to $1,500 a square foot.
What's the math on that?
It really depends on the level of equipment and.
Let's say half and half labs, half other.
I would say that if I have to throw a number at it, I would say $1,000 a foot.
$1,000 a foot times 50,000. Okay, well, there's a place to start, right? Nancy, what do you think about that?
So I agree that there's a huge opportunity today. And just think of what I said during our presentation, 2018, no private lab space in Manhattan, and now over 3 million square feet, and much of it in the kinds of buildings that Ofer was describing. So just to give you an example, when we located the New York Genome Center at 101 Avenue of the Americas, we toured over 100 sites in every borough. And one of the sites that we toured, and this was 14 years ago, nobody was doing life sciences in Manhattan. One of the ones that we toured was West 54th Street, which is where the Hudson Research Center is now, which is what I showed you, which is now full of life science companies, including the New York Stem Cell Foundation. And we evaluated the building. We found out it could sustain life sciences, and not every office building can do lab space. So it has to be in the right location. It has to have the right building characteristics. But this one did. And so we started negotiations with the landlord. The landlord did not want anything to do with life sciences or research, could not understand how you could put a use like that in their building. And so the negotiations fell apart, and we didn't go there. We went to 101 Avenue of the Americas, where Ed Minskoff had an empty building because a union had just moved out of it. And he actually put in $15 million to help us transform that building into a life sciences center. So I think there's a lot of opportunity. In 2018, when Builds Bio started, what we did was we brought the real estate community together with the academic and research community, because there's two things that need to go into creating a successful life science environment. One is you need an anchor tenant, and academia can provide that. And so if you get either one academic or a pharma, or like Regeneron, or a combination, a consortium of them, like the New York Genome Center, which is 11 of the largest New York institutions, you get an anchor tenant. And once you get that anchor tenant, then you can attract all kinds of things around that. And that allows the real estate developer to come in, and then you reach out to the tools and the supply companies that can bring resources to bear that allows you to attract companies like Sapiens. And you start to build a commercialization engine like New Lab, which has all of the tools and infrastructure needed for companies to grow and develop over time. And so at the same time that you've got this great research growing through the academic anchor tenant, you've also got a commercialization engine, which is the private companies growing and developing, or perhaps coming out of Paola's incubator and locating there. Now, and that has happened over and over again in New York City. It can happen here, and I think it will happen here. But the point is, you need to bring the entire ecosystem together and have them all put their resources into one endeavor, because density matters. You don't want, one here and one there and one there. You want this organization to be something that's connected to what's already here, but in one place. And the county of Westchester had a tour of the life sciences facilities and research that was going on in here. I think it was in March or April of this year. And I was on that bus tour and I was blown away. You got more than enough going on up here to jumpstart this cluster. It just needs to be organized properly.
Thank you. And I know, Wendy, you're sitting on your hands, and I want to appreciate that. We're going to hear about North 80 in a little while, which probably people are saying, what about North 80? Wendy's going to be in our next panel. So we're going to take a break, and we'll meet back here in about 15 minutes.
Yes.
We have a nice panel aimed at addressing an important topic: diversity, access, and celebration. Catherine Yarmula, our Assistant Dean of Clinical Skill Education and Assessment, the Director of Clinical Skills, right next door to us, is going to speak to us. Cheryl Brennan, the founder of Sister to Sister International. I just got the opportunity of meeting you a couple of seconds ago. They do fascinating work on this. Please come forward. Wendy Nadel, Senior Strategy for Advisor for North 80. I think that we met recently, and I appreciate the introduction very, very much. And I'm looking forward to continuing the discussion that we started. And Renee Pera, the president of McLaughlin Research Institute, Dean of Research of the Touro College of Osteopathic Medicine, the one that we just created recently in Montana, Great Falls, Montana. Without further ado, because I'm late, I would turn it to the podium for addressing the issues that we are wanting to hear from you. Thank you.
See if this works. There we go. I've got a fairly projecting voice, so have fun. I am, as mentioned, Catherine Imola. I am the Assistant Dean for Clinical Skills Education and Assessment and the Director of our Clinical Skills Center, which shares this building. So I'm going to talk a little bit briefly for those of you who don't know simulation, but I really want to talk about what is new and how simulation truly can enhance diversity and helping our medical providers care for a very diverse population of patients. So first off, our Clinical Skills Center opened 10 years ago in 2013. And the point of it is we want to create an innovative learning environment that can enhance and create an area for learners to feel vulnerable and to also expose them to clinical situations in a very safe environment. We have programming, which you'll see in the next page, that covers all sorts of health professions. But most importantly, I want you to know that our core values at the Clinical Skills Center are accountability, innovation, respect, integrity, excellence, and teamwork. All of these things create a synergy for any medical education program. And when I say medical education, I do mean it in its broadest sense. Not just our medical students for the School of Medicine, but anyone who comes in contact with a patient, from a healthcare provider down to the person who checks a patient in at the front desk. This all matters in making sure that we are caring holistically for our patient population. So our center has about 3,000 to 5,000 learners that come through its door each day. I'm going to talk a little bit about our big expansion plan, which is going forth in the next 6 months. But we have medical students, dental students, physical therapy students, nursing students, high school students, medical office personnel. Simulation isn't just unique to medicine. It's in so many fields. Aeronautics, arguably one of the most popular ones that started years ago, but we really can reach out to any population. We used to do mystery shopping back when I used to work at Mount Sinai and send actors into the practice to test how customer service skills. So it can be as advanced as operating on a patient in mimicked environment down to, like I said, how people are respected in a clinical practice. We have high fidelity examination rooms. So if anyone wants to take a walk over there later on a next break, feel free to check out the other side of our building. We have standardized patients, which are about 90 plus actors and educators who are trained to portray patients to our students and learners. So that is important that those scenarios are very diverse. They're highly trained. Right now, we have about 20 of our standardized patients getting a refresher on the physical examination, how to teach our students a physical examination. And that is becoming much more important in being very diverse to the ways that we talk to our patients, respecting their pronouns. If there's any trauma reminders when someone is being examined, we want to make sure that our students get access to having practice with that. So we have our teachers over there getting a refresher on how to teach the physical exam. And like I said, we are recognized nationally and locally. So simulation basically, for those of you who don't know, and I don't want to spend too much time on this, but it is the bridge between the clinical environment and real life learning. So really, what's so important is we don't put patients at risk here. So you can go over and see one of our high fidelity mannequins, which are all very diverse in skin tones finally. But the great thing about that is they can die every 10 minutes and keep coming back. And our students can actually get practice with that versus having that practice happen in the real world where there were dire circumstances, or when they delivered bad news, or they said something that was perhaps not culturally responsible to a patient. Here we can time out of that and say, okay, you learned from that mistake. Let's talk about how it felt. Let's talk about a plan, how to address it in the future, and you can move on from it. So that way, again, it's not attached to a really negative event where it really affected a patient. But that experience and that practice will have long-term effects for when they are with real patients. And it's really important that they build teamwork. So that's the other thing, is people have to work together in a diverse team of healthcare professionals. You want your professionals communicating, particularly in high-level situations. If you happen to unfortunately be in a coding situation, you want to make sure that everyone's talking and is respectful to each other, so that the student, a doctor, a nurse, whoever's in the room, all know how to talk to each other and are respectful of each other and their patients' preferences. So that is another thing. So not only do we teach important techniques here, but more importantly, we teach people how to talk to one another on a team. And we can also create a nice platform to have discussions about ethical and moral dilemmas and tensions. Certainly, there's no void of popular opinions going on in the world right now and horrible experiences and things that everyone is going through personally that we don't know about. So simulation can sometimes be a nice, safe environment for everyone to talk about those things and to share their opinions in a way that's not going to harm each other or our patients. So just like I said, it's really most importantly is we want our learners to be vulnerable. We want them to be okay with admitting their shortcomings. I also really like to explore not just social economic issues, but generational differences. We have someone who's 24 learning how to become a member of the medical team up to someone who could be 74 in a medical team. And we all have very different generational strengths and perhaps some shortcomings. And so learning about those things is really important. We have a newer generation coming off of virtual learning, and they haven't necessarily interacted with each other in the same way that previous generations do. I've noticed that our first-year medical class, they want to jump in and help so much, and they want to be innovative, but sometimes they feel very uncomfortable being wrong. So we have to teach them that it's okay to be wrong, and this is the place to have that situation and to perhaps have a in-person conversation that they hadn't been used to having for 4 or 5 years. So there's a lot of interesting things coming out of that right now, generational differences. It's also student-driven learning. So whether you are a practicing physician, a nurse, a medical student, a nursing student, it's student-driven in that, yes, there is a competency to attain, but everyone kind of gets there in a different path. And so we really support the individualized nature of learning because all of our patients are individualized. Our students and our professionals seem to think that we have a playbook on every patient, but we don't. That's why teaching communication skills can be very uncomfortable to someone because we've all been walking and talking since we were very young, and it's very personal to us. So I run a lot of courses that helps people appreciate their own communication style preferences and or biases that may have come from their lived experiences, and the fact that we have to overcome those so that we can treat everyone as equitably as possible. So this is just some pictures of our simulation modalities. What I would like to really highlight is that it took a very long time for our simulation equipment to actually represent the skin colors of our patient population. So all these companies came out with this wonderful equipment like 20 years ago. And it wasn't until 4 years ago that I started going to the sim conferences and finally saw equipment, whether it's sizes, colors, shapes, that really reflected everyone. So as all of our equipment started to age out, their computer-based equipment, those big mannequins and the little Sim Junior, they're highly advanced computers inside. Once they started phasing out, I ensured that everything that we purchased is now representative of our patient population. So that's not only important for when we're developing scenarios that do have that social, economic, or disparities that we want to make sure are highlighted. and or certain medical conditions, but it's also for our healthcare students to see themselves reflected in the patients that they're taking care of. So everything from an IV arm to our Sim Junior to other virtual equipment and virtual patients, we want to make sure that it's diverse. And so I'm really proud to say that in the last 4 years, we have completely overhauled our equipment to make sure that it is diverse. So it's not just the standardized patients. It's not just the cases. We should expect from our healthcare technology teams that they are creating items that reflect our patient population. They haven't even had obese mannequins until the last 2 years. So they're starting to make sure that we can incorporate every patient that we take care of into our technology. And they're expensive. For $100,000 a pop, these mannequins should be actively and diversely reflecting our patients. So a little bit about New York Medical College. This is online, so please don't have to worry about taking a picture. If you go on our website to NYMC at a glance, you can get a snapshot of our student population across all the schools on campus. I happen to be employed by the School of Medicine, but we have, like I said, the physical therapy program, the dental program on campus, speech language pathology, graduate studies. But 52% of our NYMC and Touro College of Dental medical students self-report being a member of an underrepresented group. So we are really actively working as an institution, both New York Medical College in partnership with Touro, to make sure that our student body is representative of the whole patient population. We are represented by 18 countries, so people come here, 18 different countries in the world. We have 46 different types of academic programs. So just again, if you're curious about New York Medical College stats, take a moment to noodle on our website and just type in NYMC at a glance. They update that pamphlet once a year. So right now it's the 23, 24 one. So it's very interesting, I always find, to go on there. So something that's also coming up in our future is we're now open 10 years. And like I said, we have so many learners coming through our doors, but I like to go out to my community and interview the needs of our healthcare providers. So Westchester County is certainly a focus, but our students actually are placed in clinical sites all the way from upstate New York out to Brooklyn. So our students do get exposed to a really wide variety of patient populations. So it's important that we have more space to accommodate the practice before they go out there. So if anyone is involved in nursing or medical school, it's a very saturated market here in New York, and we have to find placement for our students to do clinical rotations. And that is sometimes very difficult because there's a lot of other schools in the area. Nursing programs, for instance, rely very heavily on simulation because of the clinical time that they're trying to fight for. So it is inherently even more important that we can subsidize the learning here. So not just to make sure that they're prepared to go out and clinically work with our patients, but we can give them access to things that a novice learner wouldn't have access to doing on a patient. We're not just throwing a medical student in third year and saying, have fun, operate. No. So they observe, they participate, but here they can actually take a front seat. Same thing with the physical therapy students, the dental students. We just have more and more demand. And there's so many more opportunities to increase our diversity training here. So we are embarking on a 3,000 square foot expansion, which is going to have all sorts of rooms, ultrasound care, ICU, surgical labs, pediatric suites, a gynecological suite. We also want to help address the issues that are coming up with mortality rates with at-risk birthing situations or pediatric emergencies. So we partnered with the Children's Dream Foundation of Westchester to help us address those. So we are expanding. And with that comes more diverse mannequins and more diverse scenarios that we can offer to our medical learners. So that is hopefully going to break ground in about April, and hopefully we'll be up and running in about 6 months, with a much more expanded facility. So that is pretty much New York Medical College's simulation program in a nutshell. But what I always like to say is a takeaway, and certainly when there's questions for later, because I could talk for hours, is it is really magic. Anything you can think about creating, we can do. So we just had our 4th year students learning how to deliver bad news and making decisions with a patient loved one who's there talking about their wife who's brain dead. What do you do with those decisions? How do you have a decision about to take a child home or not who has a certain deficiency, which means they only have so much long to live? Versus all the way down to just basic office visits, just making sure that a learner or a medical professional is being comprehensive in their history taking. I was on the phone yesterday because my mom had to meet my elderly mom with an infectious disease doctor. And it was really wonderful to hear how open this doctor was to taking a 35-minute history. That's not always the luxury of time that our healthcare providers have. So it's really important that we also have the basic conversations of making sure our learners can ask open-ended questions that really get the patient to tell their story versus making assumptions. So you can see we really run the gamut of training. So anything, like I said, I've trained front desk personnel. So we really can do a lot with simulations to have the difficult conversations. So besides those techniques and procedures, what I'm most proud of are the realistic scenarios and the feedback, the holistic feedback that can occur when you're in a safe environment having these difficult conversations. So like I said, I welcome conversations about this, or if there's any programs that you've ever thought about wanting to create, I partner with all sorts of schools across the city on making these exciting training sessions happen. So thank you very much. And I'll pass along to my next person.
Thank you, Catherine. The presentation is open for questions, comments. Oh, fine. So I guess the comment is our next speaker is Cheryl Brennan, founder of Sister to Sister International. We all look forward to hearing your presentation. Thank you.
Thank you very much. Good morning. Maybe afternoon, nope, still morning. Delighted to see all of you, and very happy for this very intimate crowd. Let me just say, I'm Cheryl Brannon, founder of Sister to Sister International, as was noted earlier. And I am the architect of the Sister to Sister International STEAM program. And if we could move right on to the next slide, or am I accelerating? Okay, if you'd be so kind. Thank you so much. What I'm going to do is just kind of give you an overview of some of the great programming we've been involved in over the years. So first and foremost, I'm going to give you some background. Then I'm going to talk to you a little bit about some of the services and the offerings. And then I'm going to walk you through a photo journey of our 2023 summer camp to give you an idea of the various activities that we've been involved in, and of course our overall approach, which is so very important. Sister to Sister International is an organization that links women and girls of African descent globally to what we call the resources that, one, connect, two, advance, and three, strengthen us. And one of our various programs, of course, is STEM and STEAM, but we also have a pillar that's focused on entrepreneurship, pay equity, economic prosperity, juvenile and social justice, the third one, and Black maternal health. But today I'm going to talk to you, of course, about STEAM and STEM. Next slide, please. Yeah, so as I mentioned, we kicked off in 2010, and our target audience is middle, high school, college, and on to career students. So we have a wonderful pipeline, and it's really nice to see how Black and girls of color, the first of its kind here back since 2010, the first of its kind that specifically focuses on Black and girls of color. It's really nice to see how that pipeline has grown over the years. So we've seen girls, again, that are now engineers, that are now in med school, you name it, doctors, and so on. So our target audience is in Westchester and Rockland counties, to give you some background. And we really use a holistic approach. It's not just about STEM and STEAM. I often tell girls, you could be the best math or science student, but when somebody calls you out of your name, or you're not aware of a racist or a sexist issue that you approach, you need to be able to certainly handle that as well. And the main reason is that Black and girls of color are significantly underrepresented in STEAM fields. So, you know, not only are we talking, of course, about the science, technology, engineering, and math, but we're also talking about the biology, and we're talking about the entrepreneurship and all of the kinds of things that were discussed and presented earlier today. Next slide, please. So when we talk about our program, we started out as a summer camp. We evolved into a year-round academy. We provide certainly free tutoring for students in math and science, and really take a holistic approach. During the pandemic, we had a hybrid, and we're now starting to come back full time, and our summer camp was fully in person, which was fabulous. Next slide, please. So some of the services and offerings, we really expose the girls to STEM and STEAM. I often say you don't know what you don't know. If you haven't seen it, someone in your family is not in that field, you don't see yourself in that field necessarily, then it's very important that you get involved via research, workshops, internships, and so on and so on. So a big part of what we do is exposure. As I mentioned, we have the one-on-one tutoring in math and science, college prep. We provide all of these services for Black and girls of color, and they're culturally relevant, and they see people delivering these programs and these courses that look like them. So they say, well, yeah, I see someone that looks like me, so guess what? I could do it as well. So it's really about exposure and education. It's also civic engagement. They got involved with Black Lives Matter. They got involved with reproductive rights. They got involved with affirmative action, you name it. Again, we want to make sure that they are what many would say woke in some regard. Next slide, please. So now I'm just going to take you through a photo journey, morning movement. This is our session over the summer. Again, we get the girls moving. It's holistic. It's very important that that they move and understand the health benefits. Professional etiquette. Again, they get that. We also had a executive functioning for teens to help them understand, how to operate in an executive position. Telling stories through animation, key, again. Jadasola, yes. And again, the nice thing is she was an intern with us. Her mom was a member, is a member of our board. And she's now the co-chair of our STEAM and STEM work group. So my point is that we really engage the girls on a number of different levels. And in some instances, we have families, we have generations of girls that have gone through our program. Unlocking the power of emotions to really help girls tune into that. We've had a wonderful speaker this past summer. Great Warrior Women of Africa, the art project. Again, we want the girls to see themselves. So they got involved with art. Art is part of STEAM, but they also had a chance to tap into some of their culture and their history. Then, of course, we also get involved with math. They always get an advanced preview of their math and science courses so that when they go to school in the fall, they feel very comfortable. Then, of course, self-advocacy, which is very, very important. And we partner with the Westchester Children's Association on that. So we have a number of community partners leaning into your intuition, all of these very important topics that are not just math and science, but are holistic, that actually invest in the whole person that we want to make sure goes out into the world. Public speaking, we all know how important that is. Hair and skincare, we want them to be able to focus on their culture. They brought their own skin products in, their hair, and so on and so on. Then we took them as well to a special comic book exhibit, field trip. Again, the arts, but feeling it from many different vantage points. The intersection of STEAM and music with Ngazi Bell and her son. Again, just to make sure, who's the person that was referred to us through Joanne Gear. Making healthy snacks and lunch. You could be the best, again, math and science student, but you need to know how to eat from a healthy standpoint. And those are life skills. Dressing for success in a fashion show. We make it fun. We get the girls involved. We have someone that comes in and facilitates that. Then, of course, this year we took them to the science barge in Yonkers. They learned about climate change. So again, we really have to broaden the issues and make it holistic. Then STEAM Jeopardy Fun with STEAM Careers. We partner with Stony Brook University. The girls love this. This is how they're exposed to biotech and all of the up and coming fields, the fields that are certainly in demand now. Westchester County Airport, we had a couple of girls that want to be pilots. They had a chance to sit in the pilot seat. So again, all of this is very, very impactful. We host our sessions at the Yonkers Riverfront Library. We take the girls all over. This is our partnership with the Biotech Project and Joanne Gear and Juanice Mickles. We had a marvelous, marvelous collaboration at Westchester Community College, again, exposing our girls to labs and really just understanding the kind of protocols that are so important. Because when we talk about what was presented here today in terms of the incubators and the entrepreneurship, and what's going up in Manhattan and what's going up in Brooklyn, and certainly what we have here in Westchester that is on the move, we want to make sure that our girls are in a position to be a part of this as well. We partner with certainly the mayor of Mount Vernon, Sean Patterson Howard. She had a talk, as you can see, with our girls. Then our girls present the certificates so that they have the opportunity, again, to practice that public speaking. So again, a very holistic approach that's been very impactful. And then just some past events, raising a mentally healthy daughter. So we get the parents involved. As I noted earlier, they enroll in the vision and they support what we're doing. So we've done that. Just girl influencer program, the list just goes on in terms of the various activities that we, medical technology, pharmacy 411, where they get a chance to work with the pharmacists, financial literacy, mentoring circles, college prep, medicine, engineering mentoring circles. So the list just goes on. We also have a wonderful partnership with UKG, which is an internationally renowned human resource company, and we partner with their Black female affinity group. And our girls have a chance remotely to get involved with a number of different mentoring activities. So STSI is on the move. That's our story. We're sticking to it. Next year is our 30th anniversary. We certainly look forward to partnering with any of you that are interested. I'm delighted to work with Paola here, spit it out, Cheryl, and Joanne, and certainly some of you that I see in the room as well. So thank you very much, and I will pass it on.
Thank you. Thank you.
Hello, everyone. My name is Wendy Nadell, and I am the Senior Strategy Advisor for the North 80 Development. Before I get started, I just have to, wow. We go back a while when you were just starting, and you are really on the move, Cheryl, like really on the move. So congratulations for your success. So we heard in the last panel about this huge amount of activity going on up and down the east, the New Jersey, New York, Brooklyn corridor, all the way up to New Haven in terms of life science activity and development. And those of you who are sitting in the room are probably like wondering, well, like what about Westchester? Like what is going on in Westchester? So I'm really here to tell you about an exciting project on the horizon that will be breaking ground this spring called the North 80. How many of you have heard about the North 80? You've probably heard about it for about 12 years. This project has been a long time in coming. The approval process has been rigorous and arduous, and we are at the last stage of that approval. Probably that will be, we'll probably be in the ground by Q2 at the beginning of the second quarter. So what is the North 80? As you all drove in to this conference today, you probably drove, on Hospital Row, and you saw a lot of construction. That was not the North 80, but right beyond the construction is a huge wooded area, 80 acres to be exact. How do you do this? What? Sorry. Which one? Oh, this one, that one. Okay, great. No.
All right, I'm not a total moron, right? Yeah.
Is that you?
Yeah, that's me. Next slide. Okay. So the North 80 will be an 80-acre mixed-use campus, approximately 3 million square feet of space, about 2 million slated for life science and medical use. This will be state-of-the-art laboratory space, medical offices, there will be a hotel conference center, there will be at least one residential building, probably a lot more down the road, flexible meeting space for collaboration, green building technologies, and a not-for-profit STEM center, which I'm going to talk about later. Next slide, please. This is the site plan. It's a really ambitious project that will be built out in phases over the next 8 years. Phase 1, which is the first phase of the project, will be a hotel conference center, 98-unit residential building for local workforce, a 60,000 square foot lab building, and that is it for the first phase, and it will continue from there. Next slide, please. Lots of amenities. There will be a child care center, a dog park, hiking and biking trails that will be connected to the Westchester biking, the whole county-wide biking trails, health and wellness center, and a lot of spaces, both indoor and outdoor, for collaboration and meetings. It's a huge project, and it will be a real economic driver for the county. You could see here that there will be a lot of job creation and tax revenue, both to the county and really to the state as well. And most important, you can just imagine we're here. We have New York Medical College right here, Westchester Medical Center right here, the Maria Ferrari Children's Hospital right here. Down the road on one side is Westchester Community College. Down the road on the other side is Regeneron. So we're really well positioned here. As Nancy Kelly talked about, what you need is a kind of a centralized place to bring the ecosystem together. We think the North 80 will be poised to be that place that she spoke about. So I'm really excited to talk to you a little bit about this next piece. So I have the honor of working with John Ferrari, who is the developer of this project. John's a really well-respected real estate developer, mostly in Fairfield County, somewhat in Westchester County, has been around for many years. But I think in addition to his amazing real estate developments, what he's really well known for is the Maria Ferrari Children's Hospital, which he and his wife built after they experienced the tragic death of their 13-year-old daughter and realized that Westchester County did not have a facility for children. So about 20 years ago, they raised the money and got this project done. And we all know that, what a jewel that is for Westchester County and the region. So at his core, John is a philanthropist. He is very much about doing good and making a contribution. And from the get-go with this project, and when I started working with him, what he said to me is like, Wendy, we need to make this project have a soul. This project needs to have a soul. We need to figure out what it is that is going to make the most sense, where we can really make a contribution. And as a not-for-profit entrepreneur for my whole life, this is like, yeah, we can do this. And so that's what we're doing. And so when we started working together, we knew that we needed to align the not-for-profit piece with the vision for the campus, which was life science, biotech, and STEM. And what we learned over the course of about 1.5 years of listening, doing research, is that in Westchester, we have a lot of communities, and you would know this too, where we have a lot of underserved students. And students, people think of Westchester as this incredibly affluent place, but the reality of it is we have many communities in Westchester that are under-resourced and underserved in the school districts. The flip side of that is there's a huge gap in the workforce, particularly in entry and mid-level jobs. We know we can recruit Regeneron, the hospitals, recruit PhDs, postdocs, very well-educated people. But what about the students from districts that don't have thriving science programs and don't have those opportunities? How can they possibly enter the workforce? And so that's what we've been working on. And so this campus, we're right now putting together that ecosystem where we're going to be connecting K through 12 education with higher education, with workforce needs that will result in real jobs. And so that's what we'll be, if I come back here next year, we'll have a lot more to talk about with this, but that's the plan in the pipeline right now. And it really is centered on creating opportunities and access for students who don't have it, and connecting them to the jobs that are so, sorely needed in the region. Slide, please. I think that might be it. Yeah, that's it. So that's what we're up to. I think by this spring, you're going to see lots of bulldozers and excavators like breaking ground on this project. It's a huge undertaking, but I think that it will really position Westchester as the next cluster in that big map that Nancy Kelly showed us this morning, in terms of being a vibrant place for research, a vibrant place for collaboration, a vibrant place for science. And with that, I'll open up to any questions.
Thank you. And those of us who have been watching the development and the challenges and the process of the North 80 are very excited to see that it's actually seems to be happening. Okay, let's see. I'm not finding her PDF. I had it up there, but I'm not finding it now. Bear with me. It's in my folder, and it should open again. That's you? Yep. Okay. Oh, there you go. Thank you. Beautiful. I'll do it for you.
Okay. Well, thank you very much. It's a pleasure to be here today. My name is Renee Ray-Opera, and I'm coming here from Great Falls, Montana. I'm going to start with a brief vignette of my story because it will tell you why I'm in Great Falls, Montana. Okay, so I grew up in a very small town on the south shore of Lake Superior in northern Wisconsin. It was a very poor town, primarily Scandinavian and Native Americans. And the Native American population to this day has a life expectancy of about 55 to 57 years old. Ultimately, I made my way through the academic system and spent about 20 years at the University of California in San Francisco. And I was also an endowed chair for 10 years at Stanford University in regenerative medicine. Now, if you know me, you'll often hear me use the phrase, there are no paved roads between Iron River, Wisconsin and Stanford University. And believe me, that's a long story. But in any case, I ultimately decided that I wanted to devote a portion of my life to going back to rural America. And so that's how I ended up ultimately in Great Falls, Montana. Now I'm going to begin the presentation part with a quote from a recent report from the American Association of Medical Colleges. And that quote is that medical science faces a credibility crisis that directly threatens our health. And I think we inherently know that. Next slide. Now, I wanted to portray Great Falls, Montana, and so I have a couple slides here and a couple quotes from the New York Times. It is very uncommon for Great Falls to be covered by the New York Times, but it was twice in the last 3 years. Okay, and so the first quote is from an article called Montana's Golden Triangle. And the Golden Triangle is an area that produces more wheat than practically anywhere else in the United States. There might be regions of Kansas that compete. But in this quote, the author writes, ancient volcanic ranges, the Bears Paw Mountains, the High Woods, the Little Rockies sprawl in the middle of enormous wheat fields and acres of rangeland. You're small in that part of the country. So this picture right here, this is a herd of antelope, and this is out my front kitchen window. That's a car in the distance. And so you can see that it truly is, you truly feel small. And there's something really comforting about knowing that you're not that big of a deal, that we are small. We're a part of something much greater. Next slide. The other report from the New York Times appeared in October 24, 2021, and it was entitled, Where Facts Were No Match for Fear. And you can imagine there's 101 versions of this story, and many of them tracked to the pandemic. But here there was an effort to designate some land for a particular purpose, and there was no facts to some of what was said, and it was a highly debated subject. Next slide. So against this backdrop of antelope and mountain ranges and signs and information and misinformation and what people call the Montana way of life, there's this research institute. And this research institute has represented a really special part of my life, probably the most interesting part of my life. I say sometimes to people in the institute, you know, when I left Stanford, Stanford was still fundamentally the same institution. And here, what happens is that you really make an impact. We have 45 employees, about 40 of them are women, 2 male faculty members, and the facilities manager is male, and 2 male technicians. At times, the institute has suffered financially, and there was a certain point where people showed up for work even though they didn't know if they would get paid. The desire to contribute to science was so great, and the allegiance to the institute being in this town. Now, the McLaughlin Research Institute is located right here. So those scenes you saw, this is a map of Montana, and it is the closest research institute to this much of the state. The institute is under consideration, or we're moving forward to being a part of the Turo system. And Turo started a College of Osteopathic Medicine in Great Falls. I think it's one of the bravest moves I've seen. And it certainly reminds me of moving from Stanford to Montana. Next slide. Now we have institute faculty, and we are the largest, perhaps the only place in the state of Montana that has a program of such depth in neurodegeneration. Our faculty include me. I was a chair in regenerative medicine. a woman named Deborah Cabin, who works on novel therapies, Brenda Canine, who's a joint faculty member with Turo Campus and us, and then some additional faculty members who study neurodegeneration and lysosomal pathways and other subcellular pathways. Tiffany Hensley McBain, who just got her first NIH R01 grant and is also starting a company on immunology of aging. Andrea Grindelin is a veterinarian and works on chronic wasting disease. And just a key fact for you, since I'm standing in New York is the only state where chronic wasting disease has been eliminated. And so through high surveillance and culling, there is a problem between New York and Montana, and that's that there's an awful lot of animals that are not able to be surveilled. But Moses Levins is an assistant professor who has some new technology that he's patenting on ALS, or Lou Gehrig's disease. And then Michael Klingerborn works on neurodegeneration and macular degeneration. Okay. And then we have some affiliate faculty. Next slide. We recently applied for a NIH Center of Excellence grant, and we have received notice that we may receive notice, barring any unforeseen circumstances. We may receive our notice of award for this grant the first week of December. As you can imagine, just saying those words fills me with anxiety and stress. But this is a grant that is a game changer for our state and especially for this region. But the idea behind this grant is to create a center for integrated biomedical and rural health research. And the goals are to enhance research infrastructure in a unique geographical, sociocultural, and economic region of the Rocky Mountain Front and adjacent plains. The center serves two primary purposes: of increasing the number of research scientists and in advancing our understanding of common health conditions that have an outsized impact in rural communities. Okay, just to illustrate, macular degeneration changes your ability to drive. Your ability to drive in rural America is linked tightly with lifespan. There is also only one neurologist in the entire state of Montana, and so neurological conditions represent a difficulty. Montana is one of the 5 oldest states in the United States, meaning more people are over 65 than in most of the states. Okay, the center is innovative in that it provides unprecedented opportunities for talent recruitment and innovation. And I'm stressing that because I would like to offer the history of my life, and that is that unusual opportunities often have a greater mission and purpose, and I've heard that in the women that preceded me, than some of the most usual. And we think that opportunity and research needs to be distributed across the United States in order to increase trust in biomedical research. Why should people in Montana trust what's going on? There's nothing there. Why would they trust what happens in New York or California? And yet, if you think about it, and I'll just close again by reiterating the quote from or restating the quote from the American Association of Medical Colleges: medical science is creating miracles and losing trust. It has emerged from the darkest days of the pandemic with both life-saving discoveries and a crisis in credibility. It is not at all a distributed or organization. And I do think there are the opportunities. So this is my sales pitch part of the talk. I think there is opportunities for people. I hear what's going on and it's like, why not adopt programs and places in Montana? People come through in droves. Like there's 5 million people who come to Glacier National Park, which is about 90 miles away from us. And yet there isn't any work done or any investment. And I think the opportunities are really quite great. Can I have my last slide then? And these are just some pictures. This doesn't show up. This is the same mountain range outside my house. We started the first patient registry in the state last year. So there has been little or no clinical research or opportunity to be involved in clinical research. This is a open house that we had at our institute. Some of the women that are scientists, and this is a school bus from the nearby reservation. The students come quite frequently to visit us. Thank you.
Hey, especially in the upcoming potential affiliation of the Institute with Torah, I'm looking forward to working with you and you working with us in regard to optimizing and improving research endeavors, specifically for the medical school, but also at your institute so that we can provide you with the support that you need. The panel is open for discussion and please, we're gonna give you the microphone if you can identify yourself and any question is welcome at this point. Yes, please.
Hi. I just wanted to find, oh, sure. I'm Roberta Lasky, helping out with the program. I just wanted to find out, Cheryl, how do girls find out about your summer camp and your programs? Do you go to career fairs at the high school level, at the middle schools? How do they find out about you?
Sure, thank you very much. Great question. We do our outreach through our partnerships primarily with schools. So we've built up a cadre of schools that we work with throughout Westchester County. And then, of course, by word of mouth. I mean, it's unbelievable how strong word of mouth is. You have people come through the program, have success, they tell their friends, you know, the parents tell their friends, and before you know it, we have a plethora of girls. And as a matter of fact, what we've seen over the last couple of years, which is a very different trend, is lots and lots of middle schoolers. And we're really happy about that because we get a chance to have an impact on them over the years.
Thank you. Any other question? I'll take the prerogative that I have as a moderator. Catherine, in the context of STEM, and as you probably imagine, I'm not an expert in curriculum, but when we look at the curriculum, we kind of plant and seed on those STEM students to being able to groom them and continue from high school to our college, to our university, and give them a little bit of a pathway design. They can change. They may not have the MCAT, and they have bad grades later on. That's fine, that's fine. But would you consider in the summer taking some of the students that are STEM basically and introducing them to diagnosis, treatment, therapy, so that you would give them and plant the idea with them as to what is our business, when we walk into that room and we don't know what the disease is?
We actually work with about 500 high school students each summer. We work with some of the smaller groups in Westchester, but we've also partnered with a nationwide program that serves many of the students that your program speaks to in Vision. I would like to expand it to Westchester County and beyond, but so we bring these 400 students here for a day. They rotate through all other hospital systems, and we make them a medical learner for a day. So they learn how to interact with the patient and take a history. They interact with cardiopulmonary skills on the sim mannequins. They learn how to do the physical exam. We rotate them through these major stations. And what's nice about these stations and how I'd like to grow this program, which is why I definitely want to talk to you after we're done today, is that we promote that healthcare is much bigger than just applying to medical school. While certainly that is important, the skills that we can teach here is evident by all the different types of medical learners we have, is that we empower students, and they're 9th graders through 12th grade students who come through, to feel they have an autonomy, to feel when they walk away today, they can go to their parents or their friends and say, hey, I can actually do this portion of the physical exam on you, or they can go back to school and maybe initiate a club. So I've worked with a few of the high schools around here to start some health professions clubs, and they can come through here and partner. So long story long, as I some like to say, is I think we've started it, and I would love to continue to build those opportunities to bring the students here in the summer, or even partner with them throughout their school year. Because I know we always talk about the summer, but we're actually a year-round center here. And so we have programs happening all the time that we can incorporate the high school. We have some high school students coming through for one of our pediatric third-year programs. And so we actually put them in with the med students, and they learn right alongside a third-year med student. And that is so exciting because the med students love it. They see themselves in it, but then the students, the high school students love that they're on an equal footing for a moment in time, and it doesn't matter where they came from or what their background is. So I would love to make this happen more broadly than just our partnership with that Envision program.
Thank you. Any other question? Yes, please. Yes, it's coming to you.
Well, it's going online, right?
I think.
Hi, my name is Kevin Fried. I'm from Salem Oaks Consulting. I really like the topic of this, the diversity. And then I look at panel, I go, that's really diverse. So I want to know from you guys, what is the thread that runs through you all? What did you hear from each other that you'd say, hey, we need to build on that and we can work together to meet our missions?
So I think for me, I think the thread that connects all of us is education and access. I think we heard from Cheryl about her program for high school girls of color, Black girls of color. We heard from Catherine about her programs and kind of the training that goes on here at New York Medical College to train young, high school and college and medical students. What I talked about was the vision for the North 80 and the campus and what we're building there to create a STEM pipeline, particularly focused on, under-resourced school districts and students who might not have access to high quality STEM education. And I think what we heard from Renee is what she's working on out in Montana to, help train and bring education to a rural community. That's, I think, the thread.
I think another thread is that, and I heard it in the first three speakers, is a passion for the underserved. And the word underserved gets bandied around a lot, but it means that, to me, it means that to be present, to provide opportunity, and to get engaged in communities that are a little off the charts sometimes.
Thank you. I would just add that the common thread is equity and inclusion. It's kind of like, well, if you didn't get that memo yet, guess what? It's happening. And when we look at the demographics and when we look at business, We look at entrepreneurship. You can no longer say, I can do it based on the workforce all by, quote unquote, white males. It just ain't gonna happen. And guess what? If you think it is, you may not necessarily be profitable. And especially when we look at, again, the trends in population, and when we look at the overall workforce and the goals and objectives in terms of the next sector, when we look at artificial intelligence, and the list just goes on and on. So I see here the common thread of equity and inclusion, and I would hope that everyone sees the value in that and incorporates it into their day-to-day work. The other thing that Sister to Sister International has done, and we've been very vigilant about, is using a collective impact framework. Instead of just working in a silo, us, STSI, no, we are now working with various community partners on all four of our pillars. And we did the research in terms of the status of Black women and girls in Westchester County. We've got work groups. We have the pillars, and we're working together to, again, advance equity and inclusion, and bottom line, improve outcomes. And I think that's what we all want.
Can I just
Yeah,
add to that? That I really like what you just said, but sometimes I think when I use the word underserved, in a way, it almost sounds as if it's charity, but I heard in what you just said, the big deal is I think there's profitability by inclusion.
Oh, yeah.
Yeah. That there's a market and there are ideas out there that are not included. Yeah.
And I think the other theme here too is there's also I don't like to say the word hidden agenda, but there's the long stream effects that all these programs have on the things we might not always see. So for me, it's knowing that we are going to help that patients, whether they're in the remote portion of the world or they're in a very hustle bustle portion like we are here in New York. And I should have also mentioned that something that's very important that I also heard as a thread is definitely wellness for the people we're working with, resiliency and wellness. And we also work very hard to ensure that we are addressing things like the LGBTQ community as well. And I think about things that were our own personal that comes to that. I've become very open in talking about who I am, and I'm very grateful that I can be in New York to say that. I am a member of the LGBTQ community, and so while I don't represent it, it means a lot to me that I can ensure that this is incorporated into our curriculum, just as I'm sure every one of us up here has a very personal stake in what we do. And to knowing that if there is a young person somewhere in the United States and our medical learner goes and having come from a very open and nurturing curriculum, that they can make a difference on that person who is potentially 12 or 13 dealing with it, or an adult who's dealing with it, is what makes me love what I do every day.
Thank you. Thank you. I see enthusiasm in answering those questions. Thank you. Any other question, please?
Hi, my name is Supriya Jain. I'm one of the pediatric cardiologists here at New York Medical College. And I just, all the talks were phenomenal. And I think one of the other threads, which I see also, again, being an educator and working very closely with Katie, and which I agree at one of the points, which I see is high schooler mentorship, them being a mentor themselves. So, and that really sustains a lot of our programs, even at our NYMC, when we see, as Katie mentioned, if we train the high schooler then to become a mentor for another person, and then a medical student being a mentor for the high schooler, a resident being a mentor for the med student, a fellow, it just goes on. And that just sustains and keeps their enthusiasm going because they feel they make a difference. So right from all the work that Cheryl, you're doing, Katie, even Wendy, when you're doing the STEM Institute, I think, and for Dr. Para, like all of you guys are doing such amazing work, but I think I see a common thread where we are all talking about learners and educators, but when we start that pipeline early and give everybody a strong role to play and to teach the next younger one, I think, it really moves on forward. And I think that's great work you guys are doing. Thank you.
Thank you. Any other question?
My name is Ellen Bedenko. I'm from Philadelphia. I volunteer with Westchester Biotech. I'm an RN and clinical researcher. And actually, Catherine, you took the words out of my mouth because I think the overarching theme is also wellness and conquering diseases, treating diseases, and ultimately doing better to for the health of the people. So I have two questions. One question is for North 80. In your master plan, have you incorporated infrastructure for conducting human clinical trials? And the second question is for the McLaughlin Institute. You actually answered the question, saying that you just started the first registry. What are your overarching plans, and how do you think you can bring it together, bring clinical research to the residents of Montana if you can convince them to participate in clinical trials? But so have you, I know you mentioned the grant from NIH, and have you reached out to the industry and other sponsors?
I can start by answering that and saying that this is one of the problems that I think industry has with non-inclusion, and that is very, very frequently we're told we're too small to be included, rather than forming partnerships or finding ways to include people. And that means that there are entire groups not represented. So that's a problem. The second thing is I am really firmly in belief of the American Association of Medical Colleges report that I want to not, so across Montana, less than 50% of people were vaccinated. Surprisingly, in the Native American communities, more than 98% were. So there's a history there that really matters, but we need to address the problem of distrust. And I think you have to be in those communities and people have to be involved. I think to solve the problem of clinical trials, we do need to partner with organizations. It's one of the things we're excited about with being with Turo. So there's just a short fact is that there is a distrust of biomedical research, but we have found that people in Montana want to be involved. So when we started our registry, within a few days, there were 350 participants who wanted to be in Alzheimer's research clinical trials. And we started the registry because we were told that we couldn't be in those trials because we didn't have enough people, and we wanted to do a little pilot to show, hey, we can generate some people here. And remember that in clinical research now, there's two ways to do it. One, you can do kind of a survey and you need 1 million people to find a gene or a treatment, or you can do deep diving of about 1,000 people and find out what's actually happening with drugs and different things. And so we have the population, and we have a couple grants to the NIH separate from what I talked about, but we would love to be involved with industry.
And as far as the North 80 goes, it's really early days. It's probably 2.5 years before there's any building on this campus. I know that there is an interest and an intention to create a community and an ecosystem on the campus whereby that can be accomplished. But I think it's going to depend upon who moves in, who the tenants are and what their interests are, and what other partners like the medical college and the hospital here are thinking. So I think that the vision is to definitely include that as part of the community, but it's early days to have that really materialize in any significant way.
I just want to comment, we do have a fully operational clinical trial unit here in Skyline, and in the hospital. The Skyline is a outpatient clinical trial unit. but the main one is in the hospital for inpatients. So we're fully operational. We're looking forward, as we discussed last time in my office, to engaging for a larger collaboration, but there are functionally operational entities that can take multiple clinical trials. I'd like to call to the podium my dear friend, Rob Amler, who's been at the inception of BioInc. I was not born at that time, basically. Yeah. He was born, but I was not. So, but I'd like to call him to give us a little bit of a introduction of what BioInc was, is, and will be in the future in consonance with the Innovation Conference.
Thank you, Dr. Amar, and hello, everybody. I want you to know I've been with you all morning on the webinar channel, except when I got in my car to drive here, then I got it on my phone. So I heard all the presentations, saw some of the slides. I just want to congratulate everybody here. This is really a fantastic culmination of a tremendous amount of work. I can't possibly name all the players, except that I do have, oh, I left it over there. Wait 1 second. I have two great congratulations to do. One is for my good friend and former schoolmate at l'Université de Strasbourg many years ago, although I was a student long before he was. This is your very own NYMC Turo badge. So now you are official. And speaking of official, we have a new assistant dean. Katie Yamula was announced just a day or two ago. Where is our, there she is. All right, hands together for the new assistant dean. Congratulations, Katie. So how did this start? Well, you've heard this before. It started with a seed, right, or a seedling. I'm not going to take you through the entire gestation because that would be diverting from the purpose of the conference. Suffice it to say that some really visionary people, some of whom are no longer with us, got together, not in this building, but another building on the other side of campus, about 80 people who were involved in biotechnology here in Westchester at various levels. Everything from Regeneron, which was there, to some very, very small startups that some of which, like I said, are no longer in business, some of which have blossomed. And the idea was that within this biotech community, we needed a nexus. We needed a place where some of these ideas could germinate. Some of the ideas could be developed on a small lab bench, work out the solubility product of whatever it is you're working on, but not just for new candidate drugs, but also for new strains of virus, for vaccines, new apps, which, believe me, 15 years ago when you said there's an app, people would say, what is that, an AP? Is that appendicitis? What's AP? It's an app, APP, I guess, right? That was a new idea to develop some new apps. And in fact, some of our earlier client firms were developing apps that would help patients find their way to a specialty referral and those kinds of things, which did quite well as well. But also the idea of some medical devices. So these were not just wet lab benches, but also rooms in which they could develop ideas. And this gradually came together. There was great support from the leadership of New York Medical College, as well as from Touro University. And here we are. So we moved forward. We developed great space. I hope have they seen the space? They're going to see the space. They're going to see the space. Everything all the way from shared facilities, so you didn't have to buy your -80 freezer, and you certainly didn't need 8 different labs buying a -80 freezer. We had the fume hoods, we had cell culture space, and so forth, so that they could develop their ideas in relative peace and quiet, and even be able to hike across the driveway, exchange some ideas with some of our biomedical faculty, which we heard from those early client firms It was that ability to interact with our faculty that it made it most interesting. After all, you can always build a lab. You can always get a lab bench. That is not unique to a medical campus. But what was unique was the interchange with our faculty, some of which are here today or on webinar, and a new idea that I had not heard, and I thought I'd heard everything, you know, because I thought I was a big shot. But the fact is, I had not heard this term. And that was, as they developed their ideas, to contrast those ideas against market wisdom, which they could glean from the people they were talking to. You might have 4 or 5 possible candidate drugs that you could develop. Where's the market wisdom that says, this is the one you want to put your eggs in that basket? This is the one you want to move forward. I'm not a bench researcher, that should be obvious. But that connection between germinating ideas and also getting a vision for where they could commercialize and develop those ideas for the benefit of everybody was, I think, something that we were relatively unique at. So that's it. I congratulate everybody, Westchester Biotech Project, for helping us put this together. My good friend Paola, who's been fantastic at thinking through how we make a session like this work for you all. And I'm looking forward to meeting my new best friend from Great Falls, Montana. Let's hear it for Renee. And I know what it's like to live in a small town in Wisconsin. I grew up in Manhattan, so it was not me. But one of my former bosses grew up in Elroy, Wisconsin. If you know where Elroy is, where they say, such a small town, if you call somebody and you get the wrong number, you can still talk to them for 25 minutes. Thank you all very much.
So I am almost can do this without my glasses now. Well, I talked about him when he wasn't here, but I'm very happy to introduce my partner, our co-founder of Westchester Biotech Project, Michael Welling.
Thank you, Joanne. Good morning, everybody, and thank you for joining us today and sharing some of your visions and ideas. And Dr. Amar, thank you for helping us with all this, and Dr. Amler for your support as always. And you know, I was prepared to come up here and just talk a little bit about the history of our organization. We had a conversation out in the hall when someone said, I cannot wait to hear how you built the plane while you were flying it. And I was trying to think about, well, how did we do that? But something dawned on me during the last session. And I was recently, I'm involved with another nonprofit where we serve the community. And we recently did a board retreat focused around DEI, A, and B. And diversity, equity, inclusion, we all know these terms. A and B, access and belonging. And I was fascinated to learn about the concept of belonging, that it's more important, it's as important to give people the chance to learn about something as it is to make them feel like they belong in the room to talk about it, to participate in it, to pursue a career in it. And I think all of our panelists from before were all talking about the passion behind creating that sense of belonging for their constituents, both known and unknown, present and future. And that is something that, as I sort of briefly reflect upon the work that Joanne and I have been doing, is I believe that's really what the theme is we were just trying to create a sense, some space to have a conversation where everyone felt like they could belong. And while I would never pretend to say that the Westchester Biotech Project is in any way the same scope and sense of success as New York Medical College, the one thing we share in our origin story is Dr. Charlie DiComo. And yes, who was one of the very brilliant individuals early on here, a good friend of mine, and someone who, again, was one of those organizations, those companies. They had brilliant science, maybe a little ahead of their time, and just the time wasn't right for them, and he moved out of the area. And he's the one that sort of said to me, we still need to have the conversation. We got to make sure that everyone is getting together, having the conversation, making sure that the Westchester Biotech community has that sense of belonging and pushes forward. Again, we're going back 7 or 8 years. So again, how did we build the plane by flying? Well, for those of you who know me, I barely can say the word biotech. I have no training in the sciences. I was thrust into this world through a personal experience with my now, who just turned 18-year-old son, who was about to graduate high school. And Matthew, at 6 months of age, and this is a happy ending story, as I said, he is about to graduate high school, whether he wants to or not, he's going away to college. But at 6 months of age, Matthew was diagnosed with a condition that the doctors at Sloan Kettering, at Yale New Haven, couldn't figure out what it was. From 6 months of age to 10 months of age, we thought it was one thing. We were waiting. They weren't sure about the treatment protocol. Then, just by pure happenstance, at 11 months of age, I shared an observation that I saw in him, and that led them to conduct some more testing, and they identified his underlying diagnosis, which there's no treatment for other than a stem cell transplant. So we said, great, well, and we were new parents, our only child at the time, to this day still. We said, okay, great, now we know what it is. Let's get a stem cell transplant. And they said, well, not that fast. Back in 2005, 2006, the odds of finding a stem cell donor, not that easy, 1 in 6. Now you got to go home and wait, and we may never find the donor. We found a donor. We're admitted to Sloan Kettering. We start treatment. We say, great, it's going to be a round of chemo. We're going to be here for 3 to 4 weeks, and we're going to be back home, and we're going to be back to our lives. Well, guess what? Transplant failed. First donor couldn't donate again. Now we have to look for another donor, which again, the odds of finding this, they kept telling us, you got to prepare for the worst. You got to prepare for the worst. Well, we obviously found another donor, went through another series of complications and whatnot. After 143 days of inpatient at Sloan Kettering, 9th floor, Matthew was released. We're now home. Our dog cannot live with us for a year because he's still living in isolation, 32 doses of medicine. And the whole time we're saying, how are we going to get through this? How are we going to get Matthew to his 2nd birthday? How are we going to get Matthew into elementary school? Now we're saying, how are we going to get Matthew into college? And the answer to all of this is the folks in this room and all of the communities you represent online as well, who are doing the hard work, answering questions that no one else is answering. And these are sort of passions that were instilled at early ages. And we need to have programs that are instilling this passion in our children from the get-go. And we know that without it, without the lifelong commitment and passion of these scientists, of these supporters, of these sort of adjacent communities like what we try to do here, we don't get to have all these unbelievable programs that are being conducted out in Montana, the support services that are being done here at New York Medical College. So the answer to the question, how do you build a plane by flying it? You got to find a lot of people who aren't scared to crash, but share the passion to get to where you're going. And I think that's really the answer. And I could not have asked for a better partner than Joanne in doing what we've done. And when we set out, we didn't set out to create an organization. That was not what we were looking to do. I was literally trying to figure out, how do I give back to all of these individuals, organizations, communities that were collectively the reason that Matthew survived? His condition is an orphan disease. There's no foundations. No one's raising money to prevent it. I got very involved with the National Marrow Donor Program because we said, okay, he's got 2 bone marrow transplants, 2 different donors. Well, that's a place we can invest time and energy. And it was sort of in my pursuit of finding ways to give back that we were introduced to the Charlie DeComos, to the Dr. Amlers, to the people that were doing the work right here in my backyard. And rather than run all over the place and figure out, hey, how do we get these conversations going? I said, hey, we're going to figure out a way to have these types of conversations here in Westchester, and we're going to have our friends from Montana come in. We had one time the gentleman from St. Louis. We've had people from Germany fly in, and it was all about just pursuing that conversation. We never wanted to be the ones building buildings. We never wanted to be the ones, building out programs and classes. We didn't want to be the ones that were, recruiting companies here. We wanted to be the ones that were helping forming all those individuals about what we're hearing, what is needed, and how do we get out there. And I feel like it sounds like, even as I'm saying it, this like super altruistic thing. And it really wasn't at the onset. It was really a guilt that I had about having no other way to give back. But as I look in my life and I think of other projects that I'm involved in, and you look around the room, does everyone feel like they belong? Do you give everyone the chance to not just sit at the table, but have an opinion while sitting at the table, to ask questions that maybe they're thinking to themselves, I don't want to sound stupid and ask this question. And unless that person feels like they belong, they're not going to ask those questions. So I've sort of found this pattern in my life, unbeknownst to myself, creating that space for conversation. And that sort of, if I'm going to have an answer, and I'm not sure it's a good one, how do you build a plane by flying? Again, it's getting the right people in the room and having that conversation. So I always hate being the guy in between you and lunch. So I don't want to continue to sort of wax poetic here, but I think that's the best answer. And I literally came up with that since we spoke out in the lobby. But I do want to open up. Are there any questions for myself? Any follow-up questions that we can sort of just make note of and address when we come back to lunch? Or is everyone just hungry and tired of listening to us talk? Any questions, comments of any sort from anybody? Yes. Without question. And the Matthew story, while I will not bore you, is a powerful one that, again, I didn't seek out. I was thrust upon me. Again, I have no medical training, but my knowledge, I can talk about, myelofibrosis with the best of them in the world. Most people don't know what that is. But yeah, so the story is powerful. Matthew's just been the passion for myself, my wife, even with Joanne in our organization here. And just you talk about inclusivity. So Matthew, who's healthy as a horse, the one carryover from his condition, because his condition was a bone malfunction, is the best way I can describe it, caused permanent damage to his optic nerve. So he's legally blind. You'd never know it if you met him unless you threw something at him and he didn't catch it, or you asked him to read something. So another thing that I found myself looking very deep into is the idea of these hidden disabilities, and are you making people feel inclusive even if they haven't disclosed whether it's neurodiversity, whether it's a learning disability, whether it's a visual, whether it's physical. And anyone who wants to take the position to this day that this D, E, I, A, and B is a fad, that it isn't something that needs to be seriously interwoven into our society, is, I think, missing an opportunity. And Cheryl made a comment, and I'll just say that there's a study, I don't have the statistics, I can get it, but in this training, that companies that create this sense of belonging are actually more productive. And it makes sense that employees who feel like they belong, they miss less time away from work. They're not calling in sick. They're more productive. And so we're starting to see the data. Just one last thing I'll mention, even though I said I'd stop talking. There's actually a nonprofit called Just Capital, JUST Capital. And I happen to know the gentleman that runs it. Our sons shared a doctor at Sloan Kettering. But Just Capital, what they do, they basically have created a 100-point metric scale or measurement system on 100 different metrics, trying to quantify the just value of our biggest public companies. So they're looking at gender diversity. They're looking at wage equity. They're looking at how many employees at a Fortune 500 company volunteer. And they're trying to measure things that aren't necessarily in the Wall Street Journal. And they, if you go to their website, and again, I'm not saying they're 100% correct, but they are starting to show a clear pattern that companies that are just, that are treating their employees fairly, making them belong, are actually outperforming companies that don't. So again, Just Capital, just some interesting reading there. With that, am I ringing the bell for lunch? All right. Yep, sorry.
One last comment before I let you all go, just because I've been running around as usual on these events, but I have been listening in. And one thing I would like to comment on for what you spoke about, for what several of our panelists touched on, is a couple of issues. How do we address distrust in STEM in general? How do we get people included, and how do we go beyond that and make them feel that they belong where we've included them now? So starting off with the story, like Dr. Para mentioned, I started off in science. I didn't think I would. I wanted to be a writer when I was in middle school. I thought I was going to be the next Stephanie Meyer and all that. Bit of an embarrassing one. But eventually I grew very invested in my biology class. My teacher was very good at getting you invested in what they were teaching. And I loved it. But where I grew up in a small town in deep South Texas, it was not very common for science to be promoted if it wasn't directly in healthcare. And on top of that, it wasn't as common for women to be involved in science at all, especially within my community with Hispanics. So I had some relatives that, even after I was already deep into my career, questioned my mother on, why she let me come up north, because Isabel, she's going to come back pregnant. She's going to come back and have all these issues. She's not going to make it up there. She should be here at home. Why isn't she coming to a local college? And a lot of times belonging goes both ways, right? You need to have support within the community that you're trying to get into, which of course was research, but you also need to have it in your background. And sometimes the people who are not afraid to crash will be 1 to 2 relatives that support you, the one teacher that influenced you, and that has to be enough, right? You have to be able to build off of that and find your niche from there. And obviously I didn't stay in research. I ended up going more onto the communication side, but because for me, the important question became, how do I help influence the distrust, right? How do I get other young Paolas interested in science and staying in it, even if somebody tells them that they don't necessarily belong in it? How do we continue helping that to develop, not just at the student level, but at the entrepreneurial level, right? Which is what we're doing here now. So I think it's great to see how everybody has been communicating on different sides of this. And I want to thank all of you for speaking on it, for being involved in this. And now I will let you go to lunch. All righty, everyone. So now that we have our hearts and our tummies full, I would like to introduce you all to our keynote speaker, who is Dr. Supriya Jain. She is the Assistant Professor of Pediatrics and Radiology and Clinical Education Liaison for the New York Medical College, and also the Director of Pediatric Cardiac MRI, this is a mouthful, Advanced Cardiovascular Imaging for the Maria Ferrari Children's Hospital. So she will be talking to us today about some of the research that she has been working on, and in tune with everyone else's song, the collaborative nature of this research that she has been working on. So I will leave it a little vague so that you can pick up from there. Dr. Jain.
Thank you. Thank you to Joanne, Dr. Amar, and Dean Amler, and everybody else for inviting me to talk about my research. I think more than the research, I think what I was told by Joanne is how it all came about. How did we build this huge, big collaborative research network? Currently, we have 38 sites and many other sites across the United States and internationally. Do want to join our efforts. But, I've been listening to some amazing personal stories, and actually, Not even this part of the research, but all of this started with a simple story, and that it just, I never imagined it would turn into such a big study. I do not, oops, did I switch it off? See, that was a quick talk. I just moved it forward. Okay, I do not have any disclosures except the study is supported and funded by the FDA. So before I go on to the actual study, I do want to talk about, how it all started and my background. I'm a pediatric cardiologist by training, and then I trained in advanced imaging. And so I do want to talk a little bit about advanced imaging because that's the crux of the entire study and how all this tied into the COVID pandemic, then this study, and then our steps moving forward. So very briefly, I don't want to give a big talk about myocarditis, but that's one of the big, it's one of my research interests. Even as a pediatric cardiologist, that is what I have always been interested in, even prior to the pandemic. And also I've been doing a lot of research in Kawasaki disease, which is the #1 acquired heart disease in children, along with advanced imaging and other pediatric cardiology-related issues. So myocarditis affects approximately 1 per 100,000 children per year, is responsible for 0.05 of all pediatric hospitalizations. It has a wide spectrum. It can lead to, they may have non-specific symptoms versus they may present with cardiogenic shock, tachyarrhythmias, as well as need for inotropic support. And it's one of the most common causes of sudden cardiac death in young adults, and for 3 to 25% of sudden pediatric deaths. Now, all of this data is mainly related to viral myocarditis because that's the #1 cause of myocarditis, and there are certain viruses which are more common, which causes viral myocarditis, but there are non-infectious causes also, such as reactions to drugs and chemicals, which may bring about this immune response related to the myocarditis. So a little bit about cardiac imaging. There are different modalities in cardiac imaging. The most common one which we use is the echocardiogram. When you visit a cardiologist's office, this is the first thing which we do. It's an ultrasound. It's a quick and easy way to look inside the heart. And then there are the other ones are the advanced imaging technique. And since my research interest and my clinical expertise in it is in advanced imaging, CMR is cardiovascular magnetic resonance. So that's one of the things which I want to talk about. And again, CMR has become one of the most amazing imaging modality tools over the past 2-3 decades. The advancements in technology has really progressed so much further that we can have, not only can you quantify information in the heart, you can quantify the volumes, the heart function, how much amount of there's regurgitation in a valve, you can quantify flows, but now you can also do something like a non-invasive biopsy. So there's tissue characterization by cardiac MRI. So this has changed the entire thing in the past decade, where now using CMR helps look into the myocardial tissue. And so in pediatric patients, we no longer, endomyocardial biopsy is no longer done. And I'll tell you this changing practice which we have, because CMR, so cardiac MRI, it works on the basic principles of protons. And so it measures the energy which is released by the protons during the relaxation phase. So very briefly, we look at the T1 time and the T2 time, which is based on the component of relaxation. Is it on the longitudinal relaxation versus the transverse relaxation? And based on the T1 and T2 values of every biochemical tissue, you can actually identify what's going on in the tissue. So for example, if fat has low T1 and high T2, if you have myocardial edema due to the water content, has high T1 and T2. And so in a huge spectrum of clinical diseases, cardiac MRI has become the big imaging modality where you can get a lot of information in terms of fat, myocardial scar, myocardial edema, and so forth. And this technology just keeps on improving every day. It really helps in the clinical management of all our patients. So none of this is research related. It's all clinical medicine. So one of the one of the sequences which we do is called late gallium enhancement. I want to talk briefly because that's a big part of the study. Late gallium enhancement is when we give contrast to the patients, and if there is a myocardial injury or if there is your cardiomyocyte, there's an injury to the cell membrane, there is increased, you can see the myocardium, as you can see, it is enhanced. And so this picture on the left is just showing different clinical diseases where there is clinical late LGE or late gallium enhancement seen in different kind of diseases. And based on the pattern is how we differentiate between the different diseases. And all of this is correlated with actual specimens where we can correlate where there is myocardial scars or there's infarction. And the LGE is greatly used in cardiomyopathy, especially there's hypertrophic cardiomyopathy. And there is a risk stratification tool which is developed based on the LGE. I'm just going to keep on moving forward. There are some new parametric techniques which are still used in MRI. So this has changed the global practice, and the role of CMR has increased. And now I'm going to be talking about myocarditis. And so now the endomyocardial biopsy, oops. All right, so let me see. So in pediatrics, the biopsy, we usually shy away because it's invasive, there's limited sensitivity. And so cardiac MRI now has become the modality of choice for confirming the diagnosis and for following these patients. We use a certain criteria to diagnose these patients, which is called as a Lake-Louise criteria. My prior work has been looking at in the CMR techniques with patients with myocarditis. And in pediatric patients, there's a lot of data on the adult patients with myocarditis. In the pediatric populations, there are just few studies. And again, also it's related to, because it's a rare disease, you need a huge, big multi-center research network to really study these diseases. There is a well-established association of these CMR parameters with the outcomes in patients with myocarditis, that is heart failures, arrhythmias, cardiac death. Now, these, again, we are talking about viral myocarditis. All right, so how did all this get tied in? So that's a background. I am a trained advanced imager, this was in 2020 when the world changed. New York was really badly hit. We in pediatric world felt a little bit lucky because as the amount of morbidity and mortality seen by adult colleagues was not translating down to the pediatric population. So we felt that, the kids were all okay. And which is true that the kids don't really fall sick with the COVID-related issues. It's something 6 to 8 weeks later, we found that they had this mysterious illness, which was a hyperimmune. It was a delayed hyperimmune response, which was called as MISC, multi-system inflammatory syndrome in children. We didn't know what was this disease. The UK colleagues did, alert us, and soon, all of a sudden, the doors opened and all these kids were coming in. They were just exposed with COVID, either related to their grandparents or they had an infection, which was a very mild infection, but then 8 weeks later, they came in cardiogenic shock. They were sick with ventricular dysfunction. They needed mechanical ventilation. A few of them had to be on ECMO, which is we needed to rest their hearts. And so, we got, Westchester saw tons of these kids. I think to date, we have around 130 kids, which we had, that was back in 2020 and 2021. So I was right at the forefront in making and managing these patients, making the protocols. And we really didn't know what we were dealing with. This is an echocardiogram where there was ventricular dysfunction, there was coronary dilation, and we then published our findings with our initial patients with MISC, and we found that these kids were coming in very, very sick. During the course of the time, there was one of the parents who actually came up to me. The kid was really sick, an 8-year-old who was a soccer player. And she came up to me and said, Dr. Jain, is he ever going to play sports again? And I frankly didn't have the answer at that time. We were right in the middle of 2020. These kids were coming in sick. We were still trying to figure out how to manage them. The only thing which I could think about was my cardiac MRI. I mean, this was the way I could look inside their hearts. And I said, well, that was something which, I would be able to offer, except there were tons of logistic problems. Number one, these kids were critically sick. And so putting them in an MRI scanner, getting their scans done was not ideal. We would be exposing a lot of other staff to these patients. Also, we weren't sure how stable these patients would be in doing the cardiac MRI. There was very strict infection control guidelines. The scanners needed to be cleaned if these patients were put into the scanner to look into the myocardium. So it was a logistic nightmare. And so while the idea was good, it was not doable unless I came and thought outside the box and I came up with a nighttime protocol. And so I decided to set up a protocol where only it would be me and one of my techs. We would come in at 11 p.m. We would try to image some of these patients, the ones who are not very sick, were stable enough, and try to look and see how their heart was doing, how their heart muscle was doing. And this way, the kids were safe, no one else was exposed, and we could still get some basic information. And so then we published our findings, which, we would have some insight into it. And then I try to encourage a lot of my colleagues around the world and in the country and share them the protocol to say, I think it would be worthwhile to just look into the hearts of these kids non-invasively to help manage these patients. I was a part of a huge, big research collaborative network, which was studying Kawasaki disease back in 2011. And we were across 30 countries. And then we just directed our efforts to studying MISC. And since then, we have around 5,000 patients in our entire research collaborative network. But that was another place where I could share the information which we were learning. So people in New York were sharing information with folks in the rest of the country because the MISC was still not spreading to the rest of the country as yet. And so we did a lot of educational webinars to help educate people, help educate our colleagues in the management of these patients. We were involved in tons of multi-center studies where we could look into this and where we found that these patients were sick and there was LGE in 17% of these patients. Thankfully, the following research was very reassuring since these patients were sick initially, but then we would manage them, we would treat them, and then they would be fine, and they didn't have any residual defects or residual problems in their heart on the follow-up studies. So this is the kid who was actually on ECMO, was very sick, and this was a few months later. He was discharged. He was discharged home within 2 weeks. And since then, we've imaged him. His cardiac MRI has been normal. So the data has been very reassuring in the follow-up data. So now coming to how did the COVID vaccine myocarditis study come up? So I want to tell you that everything you do always, it gives you a different perspective. So had I not imaged those patients who were really sick, I think I wouldn't have that perspective when I first saw the patients with COVID vaccine myocarditis. So the COVID vaccine really helped decrease the morbidity and mortality, and it was really needed since it really brought the pandemic to a halt. It was in December 11, 2020, when the FDA issued the emergency use authorization for patients. There was a vaccination breakthrough. The first report of myocarditis post-vaccination came out of Israel, and then from the US military. In May 10, 2021, FDA expanded the EUA to include adolescents 12 through 15 years of age. I was a part of SCMR. SCMR is our international organization, professional organization for all the pediatric cardiologists, adult cardiologists, radiologists who do cardiac MRI. And I had formed this smaller subset of WhatsApp group way before the pandemic, more as and my leadership role as the education vice chair to discuss cases. And little did I know that this little WhatsApp group would explode during the pandemic and during this particular time. So in May 2021, it was the first time in this small WhatsApp group of us pediatric cardiologists, somebody shared a patient's information saying everybody was a vaccine advocate, but there was a patient coming in who 2 days after the vaccine had an abnormal clinical presentation and had an abnormal cardiac MRI, and they just wanted other people to, share their thoughts. I think within a span of 1 to 2 weeks, the WhatsApp group was exploding and everybody was having cases. And I guess as cardiologists who do cardiac MRI, we were very concerned about what we were finding on the cardiac MRI. So the best way to actually get all of this together is I try to lead the efforts, and I asked folks if they would be willing to publish a case series together, since we all had a few cases here and there. And that was the time when we actually reached out to the CDC, to the FDA. There was no safety signal at that time, but we thought it was something which we as clinical researchers could do. So we published the first report of CVAM, COVID vaccine-associated myocarditis, with 63 patients from 15 sites. We were invited to discuss our findings at the JCVI meeting at that time. And what we had found at that time, which was a surprise to all of us who do cardiac MRIs, that in the 88% of these patients, healthy kids, had evidence of late gadolinium enhancement, which was very concerning. That means there was an indication of myocardial injury. 89% of them had myocardial edema. That is when, in November 2021, I tried to initiate a larger study called Massive Myocarditis After COVID Vaccination. And we had SCMR endorsement and the FDA support. And thanks to Dean Amler, we got the FDA to hear us about our findings. And we then applied for a competitive grant, the broad agency agreement, and we then started a retrospective study and a bigger prospective study. So currently we have 453 patients enrolled from 38 sites. And the reason why I said that my experience with MISC helped understand the gravity of the situation was the MISC patients were clinically extremely sick. But interestingly, the cardiac MRI findings were actually not very impressive. They were pretty unremarkable. While the CVAM patients were very healthy, but they had a lot of myocardial injury and myocardial scar. And hence, we needed to figure out what was going on. So the research question was, number one, do patients with COVID vaccine-associated myocarditis have midterm and long-term clinical sequelae related to this complication? Do the CMR indicators, which we see of myocardial tissue damage and inflammation, do they resolve over time? And how does CVAM compare to the condition that the immunization seeks to prevent, that is MISC? And so our objectives were to look into the clinical presentation more closely, to look into the cardiovascular findings more closely, have a better understanding, and to evaluate the outcomes. So these are our sites all across the US. We have currently 38 sites. There are many more which are interested to join our efforts. A lot of international sites want to join the efforts. And so we've done an analysis on the midterm outcomes so far. And we had strict inclusion and exclusion criteria where, based on the CDC criteria, we included patients who are less than 30 years of age, and we excluded anybody who would have any other reasons for myocarditis. And so in the preliminary results for the subset of patients which we looked at, we have 333 of the CVAM patients, which we compared with 100 of our MISC patients. We chose selected MISC patients who had the cardiac MRIs, since we wanted to, those patients were one of the sicker ones and who had the cardiac MRI, and we were interested in looking at the cardiac MRI findings and correlating them with their clinical presentation. So in the CVAM findings, 67% were white, a majority of them were males. Again, during the patient the patient cohort which we had, majority of them had received the Pfizer. That was the one which was approved back in 2021, and 84% had it after the second dose. Comparing it to MISC, definitely there was the white population was predominant in CVAM, and the African American was more in the MISC. The presenting symptoms were more with chest pain in the CVAM group, while the MISC came in with more of GI symptoms along with systemic inflammation. So the clinical severity was definitely way worse in the MISC population, the ones which was related to COVID. They were the ones which had higher ICU admissions, needed mechanical ventilation, needing more inotropic support, while our CVAM group did not have as clinical, they were not as sick clinically. They had a pretty benign clinical course. The CVAM patients, 60% had abnormal ECGs, 5% presented with ventricular tachycardia. One of the patients presented with complete heart block, which did resolve. 17% had LV dysfunction, which we quantified as LVEF, ejection fraction less than 55%. It was the cardiac MRI findings again, which we found that 82% were the ones who had positive LGE compared to 17% in the MISC group. And we looked at it closely into which of the segments, this is the American Heart Association segments, when we look at the myocardium to see where we would find this late gallium enhancement. We also compared the labs in both MISC and CVAM, and while we found that the systemic inflammatory markers were higher in MISC, in CVAM, the troponin level, which is a cardiac enzyme, was higher in the CVAM group compared to the MISC group. And also in terms of the number of myocardial segments affected with LGE was higher in the CVAM group compared to the MISC. So 82% versus 16%. Reassuringly, the follow-up cardiac MRIs on our CVAM group showed a decrease in the prevalence of the LGE, although 4% had worsening, but reassuringly, again, there were no deaths or need for heart transplantation, and all the patients were doing well. So I think I'm not going to go too much into the post-vaccine myocarditis detail. We already know now, CDC already has the data that the incidence of CVAM is higher in the adolescent male group. And there are different speculated pathophysiology of COVID vaccine myocarditis. There had been some limitations because the disease may present across a spectrum of severity and symptomatology. And so currently the available data we have was, again, for the patients who presented to the hospital, so anybody who had more than minimally symptomatic disease. But the true extent of myocarditis, may be unknown. Although we do it, I think our cohort probably did have some selection bias since these were patients all coming in, and especially the co-investigators were the ones who were seeing these patients or who had done the cardiac MRI initially. So yes, but we don't know the true spectrum of it, although we hope it's not, we still think it's a very rare occurrence. These were all the sites and the co-investigators. And so I think coming down to the fact about how did all this come up, it all comes to the fact, again, we were all a part of a huge, big collaborative network where we were professional societies. I was involved in, my prior work, and the current work was leading into, guiding folks into the guidelines about doing cardiac MRIs both during the pandemic and with the vaccine myocarditis. And let me just quickly, so this is just one of we are moving on with our prospective study. We are going to look into long-term outcomes studies. These are a couple of our collaborative studies. But I think what it all boils down to is trust. I think this was a theme which we had started with, somebody had started with it. And we all had a common research question. I think we were all concerned about that, that there was something amiss. And it was truly a power of the community where a few of us got together. Yeah, I took the lead on it, and they trusted me to get things moving in a much more faster way. We published our first report in 6 to 8 weeks. And then with a more collaborative effort with the help of the FDA, I think that is when we had a seat at the table where it was important for us clinical researchers to discuss with them what we found. I think it really helps when you have more than investigators in different institutions. They are more of your colleagues and friends who has a common interest. And I think everybody was doing this research out of just a common concern of and a common question, which helped bring up all of this collaborative effort really quickly. And I think it was also helpful to be a part of New York Medical College, where we had all the resources built in, that as soon as when these cases were coming in, a lot of legwork had to be done in data transfer agreements, in legal work. We had a lot of help from the biostatistics team from NYMC. But I think it was a lot of sharing of expertise, accessing all the additional resources with the common point of how to improve patient outcome and how can we help change the dialogue, and how can we help make an improvement. So I think the key takeaways, at least of the study and the talk, was that CMR is a robust modality for the non-invasive assessment of myocardial inflammation and injury. COVID vaccine-associated myocarditis has a benign clinical course compared to MISC, but the cardiomyocyte injury, as evidenced by LGE, is much more prevalent, which needs to be studied further. The midterm outcomes are reassuring. Things are getting better, but it does warrant a more multi-centric longitudinal effort to do this. And that's where, building and maintaining the research collaborative networks is extremely important. And not only research, but I think education. So I think a part of a prospective study is also helping the sites, making them understand what kind of protocols we would need moving forwards so that everybody is doing the same standard protocols. And this is a current massive team. We have a great medical student who was very much instrumental in it. We have Dr. Draghi from the School of Public Health. We have a clinical research coordinator, and this was our data manager who helped us build up the REDCap database. And, all this was possible to get this done in Westchester. And again, this is all my collaborative team, which, as we said, it's more as friends, more than co-investigators. So it helps us to do research together. And we are all part of different studies together. And that's my team from Maria Ferrari Children's Hospital. The blue team, which we look like they're from Star Trek. Those are all my MRI techs. And that's Anthony Girardi, who, very nice, me requesting him. He never said no, driving in at 11 p.m. midnight and scanning the kids until 2 a.m. So I can't, we couldn't have done this without all these people. So thank you.
Thank you. That was great. I had a quick question about the CVAM patients. Whether or not you talked about eliminating those that had confounding factors or whatever ahead of time. I was wondering if the CVAM patients would have, or the other group might have been eliminated more often or might have more severe effects, for example, if they have some other condition that's underlying. So you're essentially, I think what I understand is you're using or you're including people that have few other factors that might impact cardiophysiology.
That is right. So interestingly, when we looked into even so our inclusion criteria was very strict on the CDC criteria. Patients had a clinical diagnosis of CVAM. And the CDC criteria is also very strict on what it was. And just because, that's why you'll see the majority of them had cardiac MRI, because that's one of the confirming diagnoses. So it wasn't that these had any other diagnosis. They were all excluded right at the forefront of. So we were looking only at the patients with a clinical diagnosis without any question, and then looking into their cardiovascular imaging findings. And the same thing for the MISC. No doubts that they had MISC. They all met the clinical criteria for MISC as per CDC. And then we were looking at these two cohorts side by side, looking at their cardiovascular imaging. So yes, so that there were no other confounding factors. Absolutely. So we looked into it to see if anybody had any prior history of even like congenital heart disease. And interestingly, neither of them, and if you'll see the data true from MISC, all of these kids are ones with no chronic illness. These are all apparently healthy, even both the MISC, the ones who got badly hit with that COVID hyperimmune response, and with these, which are CVAM, interestingly, all healthy kids. So, few of them here and there chronic, but majority were all healthy with no past medical history.
Thank you. So on one of your slides, I'm not sure if I get it right, but you see 3 different doses of the vaccine, right? And the second dose has the highest CVAM, right? What's the mechanism behind it?
Right. So we don't know. It's all still speculation. Whether, again, well, we don't know what the cause is. But even in the CDC data and the VAERS data, the second dose, again, they have one interesting thing which the FDA folks did share with us is as soon when the interval between the first and the second dose went down, was lengthened, I should say. Initially, we were doing it 30 days between the first and the second dose. And I think they increased the interval to more than 8 to 12 weeks. And the numbers started going down. We had the same information from our UK colleagues. Their UK colleagues always had a 10 to 12 week interval between the first and the second dose. And the number of cases they saw were extremely low or almost negligible. So that has to do something with the immune response where that maybe the time interval between the 2 dose interval was too short for your myocardium, or again, there's so much speculation about, is it really related to the mRNA vector? Is it related to the nanoparticle? Was it related to a particular batch, which we asked our FDA colleagues? They said they investigated it and they said no, there was none related to that. So I think there's still a lot of research going on in the vaccine front about why is the second dose way higher, the rates of CVAM is way higher. Although we did see some even in our third dose, in the booster dose, but the highest was in the second. So I think that research is still ongoing. I don't think we have a conclusive answer for that.
Thank you. So Dr. Jain asked me how she gets involved, and she can't get out now. So, but I really see this leadership, and this is in the whole context of what we're trying to do to create more spaces for growing companies. We need a leader. We need somebody who's been there, done that, who can really be the spokesperson over a period of time with a lot of support and a lot of collaboration. Is this person in this room? That's who we're looking for. Let's go on to our next panel. I'm very appreciative that a lot of good friends have been on the days today. Going back to the slides. That's perfect. Yep. And I want to give this panel credit because they're the only ones that got me their slides in time, all online. Oh, extra points. So what we're going to be talking about now is the landscape for building a career as a scientist. What are some of the programs, activities, and all that good stuff that are happening? I'm going to introduce Kevin Freert. Oops. Kevin, would you like to come up?
Sure. You said you have my slides. It doesn't really matter.
I do have your slides. Let me close this.
So I'll get started before she gets the title slide up, which they're going to spend a couple minutes on anyway. My name is Kevin Freert. I run a small company called Salem Oaks Consulting. Before that, I was at Pfizer for 30 years and did research and development. You're going to hear a lot about that. I'm also the vice president and on the board of directors for Rare New England. So Salem Oaks Consulting is actually, our mission is to empower patients to shape the future of medicine. We want to bring the patient voice to the table so that they can affect what's going on here, but they're not prepared to do that. So we're trying to educate them and bring them the tools they need so that they can be partners with biopharma, biotech, everybody who's in this whole ecosystem. Rare New England, we're trying to bring people together in the rare disease community in the 6 New England states. Which is the button that everybody kept hitting and turned it off. Okay. This one? This arrow button right there. Okay. So I didn't know who I was going to be talking to today. This is the drug discovery and development project or process as I see it, and you can see it's all over the place. I'm going to use this as the map to show you where I've been in my career. And the point is, I think Dr. Amar said it earlier, we can't see much more than 3 to 5 years out. And so when I started my career, I never knew that I was going to be here today. And I never knew I would have taken this path to get there. So I started out at the University of Buffalo Medical School as a technician in the lab. I got my degree in molecular genetics, so I jumped right into a physiology lab, and it was a great job, a great first job. My boss was the editor of the Journal of Applied Physiology. He taught me how to do science. He really made me a scientist. He taught me how to do surgery on animals. He taught me how to write computer programs and build equipment. It was a perfect first job. And that landed me at Pfizer in 1987. I skipped the whole biotech thing because there was no biotech thing. I had a molecular genetics degree. There were no jobs. Nobody was there yet. It was like Genentech and the Genetics Institute, I think, were the only 2 companies that existed at the time. Maybe Amgen was getting started. But I jumped into drug discovery at Pfizer, looking at pulmonary models of asthma, or not pulmonary, animal models of asthma in the pulmonary group. And I was building equipment again, doing computer programming, had a great time, but I kind of bumped right up the technical ladder quickly. I got 3 promotions in a row, and I was like, so what's next? Well, the next thing is, if you want to move to the next level, you've got to have a PhD. And that just wasn't in the cards at the time. The family was growing. PhD wasn't going to happen. I would have had to leave my job, which would have left us all starving. And so I moved into into regulatory affairs as a medical writer. One of the strengths I had as a scientist in the lab was my ability to write protocols and to write up the experiments so people could understand what happened. And so I took that strength and said, well, I could go into medical writing. Sure, I can do that. It was a great job. It was a great move. Had another wonderful boss. Part of what that job was all the documents that went to the FDA came across my desk. And they came from different people. And all the different pieces came together and they never matched. People didn't use the same terminology. They used different rounding rules for data. It was a mess. But what I learned from it was, well, this guy does that and she does that and they do that. And I started to put the puzzle together of how does this place work? Who's doing what? What are the responsibilities? And people notice that. And they asked me to they asked me to educate our commercial divisions about how R&D works. And I built a simulation which squeezes 15 years of work into one day, a team-based competitive game. About 40,000 people have taken that simulation now. It forces you to see the impact of your decisions minutes later rather than 5 or 6 years later. And so people were learning the process very, very deeply. To do that, I had to ask all the senior VPs of research and development, okay, I've got 15 minutes to simulate your career's work. What's important? And they would give me a couple points and I build it in. But what's important is that I built a network of all these VPs. I had a bunch of important people that thought I was doing good work and I understood their business somehow. And so the job of chief of staff for the head of worldwide development came up and they basically anointed me. They said, Kevin, you just want Kevin. Don't interview anybody. Don't post this job. And I was given that job and I had just gotten my MBA. So I was all excited. This is cool. I'm in a business job. And I was bored. It's a really cool sounding job. Wow, you get to do all these things. I basically was handling the strategic agendas for our meetings when we come together and try to figure out what are we going to do for the next few years. It used about 30% of my brain. So I asked my boss, what should I do, Craig? And he said, go add value. So a friend and I invented something called Pfizer Research University. And what Pfizer Research University was, we took all of our staff and turned them into students and faculty. We had them teaching each other what they do. This whole process that I showed you before, it's really complex. When a drug gets approved and you have the big celebration, you do a team picture, there's 500 to 700 people that can legitimately say, I had a hand in that. So they don't know what each other does though. I was the guy who was sitting at that medical writer desk seeing it all. They're just all down in their cubby holes doing things. Pfizer Research University broke that open and had them teaching each other what they do. Unfortunately, fortunately maybe, Pfizer got into merger mania. We had a whole leadership turnover and they didn't understand exactly what PRU was trying to do. And we got slowly dismantled. I found myself in human resources for about 3 years. It's great. I picked up some credentials in change management, leadership development, team effectiveness. And I was like, but where's the science? Where's the medicines? Where are the drugs? So I jumped into project management. And project managers basically orchestrate this whole process. And what they did here is all this stuff I had learned over the years, now they were holding me accountable for it. I had to deliver. And for about the last 11 years of my career, I was leading drug development teams, most of them in the later part of the clinical development process, one of them into commercialization. And I loved it. I actually ended up on the leadership team for global project management when I retired. And just before I retired, I thought I was going to become a pastor, just to really take a left turn here. But just before I retired, we had a patient come in and talk to us, a patient with Parkinson's disease. And she sat up on the stage and told us all about her Parkinson's, what she has to do every day to deal with it and manage it. And then she told us specifically what she was doing at that moment to manage her disease on that stage. She was sitting. She had somebody next to her nudging her saying, okay, shut up now, move on. She had specific things that she had written down and she read things to us because she didn't know what her Parkinson's was going to do that day. So she did all that introductory stuff and then she just tore a hole in us about our clinical trials and all the things that we were doing wrong. And she wasn't whining. She was critiquing. She was speaking our language. She was telling us exactly what we did wrong. And I was just talking to Dr. Para before. Parkinson's is just this multifaceted disease. Everybody's is unique. But if you ask any Parkinson's patient, are you on or off today? They can give you the answer. They know if it's a good day or a bad day. She said, you never ask that question. It's the most reliable data you're going to get in this heterogeneous disease, and you never ask the question. I chased her down afterward and said, okay, you talked our language. How'd you do that? And she happened to be an insider. She worked for a company that developed patient-reported outcomes. But the person from the Parkinson's Foundation who accompanied her said, but we train our patients to be these kinds of advocates, to engage with companies. And I asked, well, what do you teach them? She said, oh, we got 2 or 3 slides about clinical trials. Well, gee, I thought, I've got an idea. I'm going to start a company when I leave, and I'm going to do what I was doing within Pfizer, teaching people how this thing works for patients, families, advocates, and nonprofits. And that's what Salem Oaks is. I don't know if you guys know the word Salem means peace. It's like shalom. And in my backyard, I've got a 600-year-old oak tree, and it's really strong. So I've got peace and strength in my name. I also do a couple of podcasts, and if you'd like, I can share a card with you and you can listen to my podcast as well. The one thing that happened to me is I got really involved in rare diseases while I was still at Pfizer, and I love it. We were talking about diversity and underserved populations. I do a podcast with a guy whose son is one of 10 in the world with his disease. He's not served at all, let alone underserved. There's no service there. He's barely treated symptomatically. His son just celebrated his 5th birthday. It's great. What we've done in Salem Oaks, we've tried many different ways to get our education out there, but we've recently started something we're calling the Atlas. And what it is this vision in my head of all these things I can teach people, but I can't just put it out there. They can't swallow it all at once. I'm going to have to do it as they need it. And so we're doing these little micro-learning things. It's kind of a subscription service. I talk to my members and say, what would you like to learn next? Where are you now in your research? There's something you have to know about rare disease patients and families. They're out there doing the science. No one else is doing it for them. There's someone who we never speak about here when we talk about academic institutions and industry, organizations. And there's people out there starting their own companies, starting their own high throughput screening, starting their own facilities. And so that's who we're trying to serve, and that's how we do it. So I'm going to end a little early there, but.
Thank you again for inviting me. I've been asked 2 particular questions. How is your institution preparing life science business professionals? And what challenges do you see ahead for them? So being from an educational field first, right, I took a look at, okay, what are the top science fields that are in demand right now, according to Indeed.com. Okay, so these are sort of the fields, right, in the life sciences that are in demand, as you can see. Okay, and I'm sure some of you are in that field, right, or maybe trying to approach in both fields. And then the next question I asked is, okay, what are some of the pathways that these scientists can take, right? So what areas can you find work in, so to speak? Okay, and here are just some of them. The majority of them are in research, whether it be in academia, in government, in the public sector, commercial development, etc. And you also have ones such as science communication and publishing. Okay, and of course there's always the policy, the legal and regulatory affairs areas that scientists and life scientists can find their jobs in. So what are the biggest challenges? Because before addressing the question of how does Touro University Graduate School of Business, right, address the challenges, right, address this, is what are these challenges that life scientists face? And so in doing a little research, these are sort of the top challenges, right, that we see that life sciences face, or researchers face, right, or scientists face, right? From technology to task management to obviously trend analysis, right? But also communications and dealing with individuals, right? Dealing with staff and team, okay? And what are the best practices, so to speak. In addition to, of course, operating costs, profitability, trend analysis, marketing, et cetera, all of the business aspects of it. And this is what they found to be most challenging. So how is Touro Graduate School of Business addressing these challenges? Okay, we sort of have a two-pronged effect. Well, three if you count the Career Resource Center. But we sort of have a two-pronged effect. The first one is through our co-curricular program, interdisciplinary program. And the second one is that we have 2 degree programs that are specific, right, in the health field and, of course, in entrepreneurship. So please allow me to go through each one. Okay, so for co-curricular programs, we have 2 co-curricular programs that we support and sponsor. It is available to all students at Touro University, whether they're undergraduate, graduates, they're in part of the medical school, the pharmacy school, or any of the schools that are all the 35 schools within the Touro University system. Okay, it's all free to the students. Okay. The first one is the Moonshot Scholars Program. That usually takes place in the fall semester. It's about an hour, okay, for about 10 weeks. 5 weeks, we sort of go over a book called Bold, okay, that we asked our students, right, to purchase a couple of dollars, all right? And we sort of go over the principles. In other words, what is your moonshot? These are students who are maybe thinking about, right, some sort of entrepreneurial program or sort of their, if they had a choice, what would they do, right? What is their moonshot? And so 5 of the sessions, we sort of go over the principles, right, in the book Bold. And then the other 5 times, we actually bring in guest speakers who actually are in the industry and they're actually, talking the talk, right? Or talking the walk, whatever. How is that saying go, right? Walking. And then for those, after that is over, in the spring semester, for those who really want to go further and say, okay, I've developed my, my idea, my concept, right, for this business, right, that I think I really want to pursue. Then we prepare our students for the business plan competition. Okay, we take them through all the steps in building, right, their business, so to speak, and building their business plan, all the components of the business plan. We work with them in solidifying their elevator speech, so to speak, right, or their pitch to investors. And if they want to, they don't have to. Again, this is all voluntary. It's all free of charge to the students, but we help them through it. And if they want to, they can actually compete in the business plan competition according to their tracks, so to speak, that they are pursuing. We sort of switched, we're in part of the Mid-Hudson Valley now this year because we're in Times Square. We've moved to our new location. So now we're part of the New York City Business Plan Competition. And by the way, we're hosting the New York City Business Plan Competition this year. So we're really kind of excited about that and see how that goes. But as I said, we've been in it for about 3 years, and we've actually had 3 winners. So we're really proud of our students, for competing, for actually bringing their idea, right, to the forefront. And if you do win, not only do you get advice, but you also get some money as startup. So these are our co-curricular interdisciplinary programs. And moving on to our credit-bearing programs, right? We have an entrepreneurship program at, a graduate school of business. We have an MBA with a concentration in entrepreneurship. So why entrepreneurship? Well, we have expert faculty. All of our faculty members are really not only book savvy, but they're also professionals. They've been working in the field. Okay, hands-on, experience, and then of course, our programs. Okay, so our 42 credit program MBA that we have, and our specialization is, or one of the specialization is in entrepreneurship, where you can actually help those who want to do it in terms of, seeking venture capital, angel funding, and all the fun parts, right? The more mathematical part, and the marketing aspects, right? The whole business aspect, right, of starting your own business. These are just a few of the courses, okay, that make up our entrepreneurship program. And if you notice in some of these titles, they are all fulfilling those life science challenges that I showed you in the prior slide, right? The challenges that life science entrepreneurs, right, are facing. And the courses that we offer are sort of to help, right, bridge that gap, to provide that information, right, of the challenges that they will face. Now, we also have a degree program in healthcare management. For healthcare management, we have 3 programs. We have an advanced certificate. These are credit-bearing degree programs, right? That later on, if they choose, they can transfer all of the 15 credits, 5 classes into the MS or the MBA, even at a later stage in time. We have an MS in healthcare management. So if you really want to do a deep dive into the healthcare field, right? And then, of course, we have the MBA in healthcare management, which gives you both, the breadth of business and the depth, right, of healthcare. And again, these are just some or a few of the courses that are included in the healthcare management field that again address some of the challenges that are faced by life sciences. And one of our latest that we're kind of proud of is that we also offer Six Sigma, and which leads to Six Sigma certification, which is very much in demand right now, and it's part of the curriculum. And students can also take it not for credit. So if you just wanted, you're in the field and you said, hey, you know what, I've heard about this Six Sigma. I'm not sure exactly what it is, but I want to do it. I want to obtain certification in it, right? You need to know statistics, okay? but I just sort of look. But we do have a prep course for statistics that will help you out right before you do that. So we do offer both that course for credit and not for credit. I'm going to stop right here because I think I addressed the 2 questions that were posed of me. I want to thank you. If you have any questions, issues, here's my information. I'm available. Thank you.
Thanks, Joanne. Can you hear me okay? So thanks for the invitation to speak today. My name's Avram Kaplan. I'm currently the Associate Provost for Research at Pace University. But the story I'm going to tell you today came from my work primarily at the City University of New York, which kind of brought me to Pace, and I'll tell you about that at the end. So I started my independent scientific career as a cell biologist studying protein folding and protein degradation in a yeast model system. It was fun. I had grant support from NIH, and I was at Mount Sinai for about 15 years. But in the end, as Kevin said, your life changes, and I felt if I had to publish one more paper, like I'd been there and done that, and I felt bad about that because I had these NIH grants, but I was also concerned about my impact and how many people were reading my papers. And sure, if you get cited 100 times, that's fantastic, but you use up an awful lot of resources to get. So I was given an opportunity to move to the City University of New York into their central offices as an associate dean for research. I knew nothing about administration. My last slide is what I've learned and how to do this. And to move my lab to City College so that if it didn't work out in administration, I could go back to the lab. So I moved to CUNY into their central offices, first at 80th Street, then at 42nd Street. And I learned a great deal about science, about people, and about higher ed that also concern me. So CUNY is huge. It's a multi-billion-dollar enterprise. I think their current budget is about $4 billion, 250,000 undergraduates. There's 30,000 graduate students. It's spread across 11 senior colleges, 7 community colleges. And one of the things I noticed going to CUNY from Mount Sinai was, first of all, most of CUNY most CUNY colleges are teaching colleges. They're not really research colleges. Hunter, City College have outstanding, high impact research. But most of the colleges are teaching colleges, especially at the community colleges. Nevertheless, all the faculty are governed by the same union contract that governs their tenure and promotion, so all faculty are expected to do research. But here comes the second issue. All faculty are hired based on their research and scholarship, but their day job is teaching, and most faculty aren't actually trained to teach. You have to learn on the job. So that creates a whole bunch of issues for students, not least of which are the achievement gaps for those students who are underrepresented in science. Minority students who are underrepresented in science, Black and Hispanic students, achieve way less than white and Asian students in the CUNY system over multiple years. And I'd looked at that data, and it's really quite stark. So the students in STEM aren't really being served for achievement by the faculty, and the faculty are hired to do a job they weren't really trained for. And so where does a dean of research come into this? So I became interested in undergraduate research as a way to bring the scholarship of faculty more into the classroom. That if you could bring research into a classroom setting, the faculty would be able to carry out their scholarship like they were trained to do, but in an environment that would benefit the students and bring real, authentic research, as we described it, into the classroom. And we were fairly, I guess, pedantic about this in the beginning. We were more dogmatic than pragmatic about what authentic research meant. But nevertheless, I was at the central offices where there was money to fund small grant competitions for $5,000, something like that, on a competitive basis. And then we asked for solicitations from faculty across the system, and there are thousands of faculty members across the CUNY system, to write a proposal to propose how they would put their authentic research into a classroom setting. And we would solicit these applications and then fund them and sort of build a community of practitioners who are teaching in this different way, and hopefully it would make a difference in student outcomes. And that was the general idea. So I'll tell you sort of two stories that came out of that. The first story is a project that I became involved in personally with a professor at Brooklyn College, Theodore Muth, who's a microbiologist, and our postdoc who we hired together, Jessica Joyner. And together, TR and I wrote a NSF grant to take his ideas, which was to do metagenomics in a classroom setting in Brooklyn. So he was having the students go out on the street, the subway, the parks, wherever, collect whatever they wanted to collect with a cotton swab, come back and actually identify the microbial communities and to see whatever they could see. And they found all these pathogenic bacteria surrounding Brooklyn hospitals. There was a lot of interesting science that they were doing. It's simple science to do because it's just isolating DNA, PCR amplification, then you send the PCR products out for sequencing, then they come back so you can do some data analytics. It's a pretty simple idea that he was doing at Brooklyn. The question was, how do you scale it across the CUNY system? So TR had the science, but I had the administration from CUNY Central to leverage at scale. And that was my job. My job was to help to scale the project. And I was interested to see if I could scale the project, to convince other people to get involved. And we did that. We were able to scale this project across the CUNY system. We had thousands of students running around doing this work. We had millions and millions of DNA sequences, new bacteria. It was really fabulous. CUNY had, at the time, a very large computer to compute all this, and we hired a postdoc to help train the faculty in doing data and analytics. And so it was a really good project, and we were able to scale this nationally through the American Society of Microbiology, allowed us to run workshops at their conferences. And so this was a great example in how we could take one idea of how to integrate research in the classroom and then scale it up. So this was this was good. This is what NSF was really interested in. How do you scale it? We got a second NSF grant to try and figure out how to solve the data analytics problem, because most faculty who teach are not computer scientists. But in order to really work on metagenomic data, you need to know something about computer science. So we had a second NSF grant to do that kind of work. So this was good. We learned how to scale. We got more faculty involved in teaching students research and having them perform authentic research, but was any of this of value? Okay, so how do you assess this? Now, it turns out that these small grant programs aren't big enough to assess properly, and we never really got to assess the authentic research experience in microbiology project properly. But I did start another program at CUNY Central that we were able to assess. And I think this is where we were able to really see the value to students of research and how to resolve the achievement gaps for underrepresented minority students. So CUNY, being a multi-billion-dollar enterprise, there are often millions of dollars available to run projects if you have a good idea. And it sounds weird coming from Pace University, where there isn't that kind of money. But the city of New York funds all the community colleges. So the state funds the senior colleges, but the city of New York funds the community colleges. And Mayor de Blasio at the time actually was interested in STEM education at community colleges, gave us 50 million to do advising. And some of that money, small amount of that money, came to me to develop a research program at the community colleges. So I was able, with a team of folks, including a sociologist, to set up a program at the community colleges where the students were paid, I think, $5,000 a year for a year-long research project with a faculty member at the community college. And at CUNY community colleges, the faculty do research, because that's the only way they can get tenures to have some kind of productivity. So we developed this program. It was a year-long STEM research experiences program, and then created all these add-ons into the program, like boot camps in coding and summer symposium. There were travel awards. We let the colleges have some kind of independence in how they recruited students. But importantly, we were able to use the power of CUNY Central to assess the program using propensity score matching. So for every student enrolled in the program, we were able to search the database for an equivalent student based on 17 different metrics that did not enroll in the program, but nevertheless had the same grades, gender, financial background, you name it. We were able to figure out an equivalent student. So we were able to do some sort of assessment. And we found our results. So what our results showed was that students who engaged in a one-year research experience were much more likely to get a degree in STEM, to transfer to another college and complete a degree in STEM, a bachelor's degree. But most importantly, the students who transferred within the CUNY system transferred primarily to the research-intensive colleges of City and Hunter. And for the students who transferred out of CUNY, about 70% of them went to R1 schools compared to the comparison group. So this to us was a big win, because it said that the research experiences of the students had a direct impact on their outcomes. And more importantly, although it's not shown here, we actually leveled the achievement gaps between Black and Hispanic and Asian and white students at the community colleges. So there was no statistical difference between any of those groups in when they got their degree and where they went to afterwards. So what have I learned doing this? And part of my motivation for getting into this kind of work was to take what I'd learned as a scientist, as a researcher, and how to apply that to administration. And so, okay, so what I've learned is this, is research administration needs a research mindset. What you learn reviewing grants for NIH and NSF and papers really helps in designing programs, small grant programs or conferences yourself, and that your credibility derives from prior success. So at PACE and also at CUNY, I have credibility because I'd achieved at that level. And you also need a compliance mindset, and I had to learn that. And you have to be on top of compliance as a research administrator. But I also know I had to learn new skills. And I guess like many people, became involved in laboratory research because it did not involve interacting with people. And but you really have to learn how to do this to be successful. And I noticed going to conferences through the years that the people who were successful in science, really successful, who were my peers who became very, very successful, were also very socially successful. And I saw how socially successful people operated, and that meant a lot. And this is something that we began to incorporate into our CUNY Research Scholars Program, is how to develop social skills. But as an administrator, you need to develop empathy, at least I did. And you also need to learn administrative skills. You have to understand communication and marketing. Mail merge is your best friend. And these are skills you have, these are learned skills that they're not natural to scientists. And we like to communicate to a very small niche audience in the language that we develop with our peers. But as an administrator, you have to learn how to communicate more broadly, and you really have to learn marketing. And your impact can be broad. And I think you have to think big as an administrator and think about outcomes. And what I really like about it is that you're directly affecting the lives of those around you. And that, to me, is actually worth a lot more than the papers that I'd published in the past. So I'll leave it there. Thank you for your attention.
Hello. I'm glad you talked about money a little bit. With the Association for Women in Science, we had a scholarship program, and there wasn't a whole lot of money in it. And my first thought was, oh, let's put all the money together and give it to one person. But then we found that the AAUW had done a study that the earlier a scientist gets any recognition, and if there's one penny connected to it, their income is much, much, much higher than anybody else. So, and I like it that you bring up social skills. Juanice Mickles, bring us home, girl.
I'm going to certainly try. Hi, everyone. My name is Juanice Mickles. I am the former Associate Director of Westchester Biotech Project, and I'm also a current senior at Berkeley College studying business administration and management. So I want to give you a little snapshot of earlier in my career because it's a bit unique to what you've heard so far. So here's an introduction, just talks about some of the things I've done early on in my career. I worked in private equity as an assistant, not very inspiring at the time, so I moved on. Through a friend of mine, I found out about a position in film advertising, and I worked my way up there. I started at the front desk and I ended as a senior account executive. So that was a lot of fun. But as life happens, the company merged with another company and we were all let go. So that was my first taste of being laid off. Fortunately for me, I also had friends in event marketing and other companies, and I was able to just pivot my career. Fortunately, again, I stress that because not many people can pivot their careers, but I did, and I worked in event marketing and promotions for 4 years. And that was quite interesting because I was a consultant. So, of course, when you're in a consulting capability, You don't have other benefits. So after a while, I said to myself, well, maybe I need to do something different. Let me see what else is out there. And you'll see a common theme and thread with my entire presentation, which is networking. So fortunate for me, again, I heard of a position at a commercial real estate firm. Now I had no experience there, but I said, well, you know, they're offering me an entry-level position. Let's see what happens. And it was at that company where things changed for me. After a while, after a couple of years of just doing administrative tasks, being quite bored, but not really knowing where I wanted to go, I didn't want to be a broker. You know, my experience was in marketing. So what was I going to do here? And their marketing department wasn't that robust. So I really didn't know what to do. And this is where leadership comes in, because our department changed leadership, and that leader really changed the trajectory of what I was going to do later on. That was the first person to say to me, to come to me directly and say, I don't know you, but I'd like to get to know you. What do you like and what are your interests here at this company? And that was the very first time, honestly, in my life that someone said that to me. And we talk about diversity, equity, inclusion, access, and belonging. That was the epitome of that moment where I felt seen and heard. Someone in leadership actually came to me and said, who are you and what are your interests? And that changed everything for me. I said, well, I have a background in marketing and events. And he said, great, what can you do to reinvigorate our young brokers who came to this industry just thinking they were going to make a ton of money and that they didn't need to do anything else? And I said, well, thanks for asking, because I had plenty of ideas before that were shot down by the previous leadership. So what I did was, before enrolling in college, I created a broker and mentorship program. I said to myself, well, if we want to show that these young guys are not just about making money in a sales capacity, what's the best way that we can show that they're also able to give back to their community? I was already volunteering with several organizations, nonprofit organizations. Let me make a phone call. So I made a phone call and I got those young guys out in the field talking to the community and serving food for underprivileged people. So that was a successful moment for me, and it also felt good, and it made me feel valid as an employee at the company. The other thing that I did was a volunteering initiative, kind of the same principle, where I reached out to another organization and said, hey, do you guys need any help? And we were able to continue doing that, and the formula worked, and now we had, you know, more eyes on these young brokers, and now the company was being seen. I was also, as you could see, enrolled in college, and the way that that happened was I was just thinking to myself at that point, what else can I do for me personally? I didn't have a college degree. I got through life and up until that point, really just networking and seeing what was out there. I'm naturally curious, but enrolling in college was the best thing that ever happened to me. I started out as a marketing student, and then something changed. The pandemic hit. And as you all remember, I'm sure many people got laid off. It was just utter chaos. I was laid off. I was actually, fun fact, I was laid off on my birthday. And as you can imagine, that was really scary. It was a little depressing, but I thought about everywhere I've been since then, and I said, well, everything that I've done has come from me being curious and me knowing there's something more that I can do. So the pandemic, layoffs happened. I knew I wasn't going to get another job in commercial real estate as a marketer, where my job was bringing together large amounts of people. That wasn't happening. Fortunately, I was already enrolled in college. So I changed my major to business administration and management. So let's fast forward to spring of 2022, when many things changed, more changes. I love the changes. Through my school, and I think this was a mistake, I saw an email saying, again, remember, I'm a business student. I see an email saying the Westchester Lab certificate. What? Why did that come to me? I'm not a nursing student. Westchester Biotech Project. Thank you, Michael, and thank you, Joanne. We're doing a pilot program, and it was a 2-day lab certificate program being held at SUNY Westchester. Now, I could have just deleted that email or said, no, doesn't pertain to me, why would I do it? But then I looked at the details and I got really excited. I was going to be in a lab coat, in a classroom full of other, I'm sure, professionals or students who were science majors. And I was going to be able to participate in a science experiment. That was so exciting to me. And I immediately thought of back in middle school when I loved science. It's almost like it hit me. Oh yeah, that memory. I loved science back then. But guess what? I was terrible at math. So I moved on and I did other things. And Joanne and I have talked about this at length a couple times. We found that middle school is that time in a kid's life where they either talk themselves out of continuing their education in science or they continue pursuing it. I didn't have resources when I was in middle school. I didn't have Westchester Biotech Project. I didn't have Salem Oaks. I didn't have Sister to Sister International. Back then, I didn't have those resources to take me under their wing and help me with my math issues. So I moved on then. So going back to summer of 2022, that's when I took the course with Westchester Biotech Project. And I can honestly say it was the best 2 days of my summer. Thank you, Kevin, because Kevin was one of the speakers, and your talk about the drug discovery process literally had me on the edge of my seat. Kevin was another huge inspiration and helped me continue in my quest and my journey. So another thing that Kevin did was he sent me to my first pharma conference. Mind blown. I knew nothing, and I came into it letting people know that I knew nothing. And there's something really interesting about saying, I don't know, and I think someone else spoke about this earlier. I found that people were more interested in me when I said, I don't know this, than when I tried to pretend that I knew something about pharmaceuticals. So that was an interesting thing. So thank you again, Kevin, for that. Now, when I did the class, the Westchester Lab class, afterwards, Joanne and I had a conversation, and I must have been like a puppy dog to you because I was so excited. And I just wanted to know more about this class. This was a pilot program, but are you doing this next year? Can I get involved? And through our conversations, she allowed me to work with the company, first as a researcher, right? And then eventually I became the associate director. So, steps toward career progress. Simultaneously, because I'm a simultaneously type of person, I was a student. I was associate director with Westchester Biotech Project. Someone in my network called me and said, did you know that there's a program called Life Sci New York City internship? And I said, no, tell me more. I like to know things, tell me more. And she sent me information. So Life Sci NYC is an internship program that was developed by people at the New York City Economic Development Corporation. And I had a conversation. I love having conversations with the executive directors. Had a conversation with someone and they said, we'd love to have you on board, you know, to send us a personal statement. And all of the things that I told you prior to what I'm saying right now was in that personal statement, and I was accepted. While that happened, the organization, LifeSci NYC, was the sponsors for my host company, which was New York Bio. So New York Bio wanted me to, and think about when I tell you what this was that they wanted me to do for my summer internship, no pressure. They wanted me to write a DEIB guidebook for small biotech firms. Now, as you heard, I didn't have much experience in the life sciences except for what I did the past year, but I was up for the challenge. And I believe that it's on the Westchester Biotech Project website, the DEIB guidebook that I wrote. So that was over in the summer, this past summer. Simultaneously, again, I was contacted by a friend of mine who's the Chief Diversity Officer at Columbia University. And she knows that I was interested in figuring out how I can contribute to my community's health. I live in Morningside Heights in Manhattan, and I see what's going on in terms of healthcare. I see and I hear some of the comments about trust issues in my community, and I wanted to know more. So I applied to be a community health worker, which was a free 8-week program, 6 weeks virtually and 2 weeks on site. That ended a month ago. So I am now also a community health worker. I did my first healthcare screening at a church in my community. And Kevin and I were speaking about this earlier. One of the things and one of the common threads when it comes to doing community work is that you hear things like, we didn't know that this existed. We didn't know that we could get these types of services. What else can you tell us? How else can you be a resource to us? And to me, that meant the world. That meant that people are actually interested in knowing more. They are interested in their own health. They just don't know where to go and they don't know where to look. And that's where we all come in, right? We're all a resource, every one of us. We're resources for community health. We're resources in the life sciences industry. So the last question I was asked is, what's next? What's next for me? Graduation. I have 1 week of school left, so I'm in finals right now. I will graduate with honors, which I couldn't have said the first round of college many years ago, so I'm happy about that. Community health fairs. I need to complete 2 more in order to graduate. By the way, the graduation will be on the roof at Columbia University. So I'm looking forward to that. I will be doing many more than just 2 community health fairs because I absolutely love it. And networking. I love speaking to people. I love talking and hearing and sharing ideas. I'm always trying to find out someone else's story. I think we all have really interesting stories and I'd love to hear all of them. I'd love to be able to collaborate and see what else, what do you have going on and how can we work together? One of the things that I'm really happy about was being able to connect 2 companies together. I love talking to people and I like to make those connections. And mentorship. Simultaneously, over the summer, someone called me and said, I have a classmate who is having some visa problems, which means that she can't get an internship. She wants to go into commercial real estate. So going back, I won't go back with the slides, but remember that leader I talked about in commercial real estate? I called him. And I said, there's this woman, please just look at her resume. He said, we've already chosen someone. And I said, well, do me a favor. I'm gonna text you her resume. While you're at dinner with your wife, please just look at her resume. And the next day, they called her and they interviewed her and they hired her as their summer intern. So giving back is something that I'm really passionate about also. You know, at one point, I'm asking for help, but it felt really good to be able to help someone else at the same time. Thank you very much. Here's my QR code if you'd like to keep in touch, and I'd love to talk to you. Thank you.